Hello guys, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. I hope I am audible, visible and the presentation right behind me is also visible. Please let me know in the chats. I hope all of you are doing the quiz well. Yes, very nice, very nice. I can see a lot of questions in the chat box. So that's a very nice thing that you can do, right? When you're waiting for a class, you can ask questions to each other from the previous class. Now, molecular basis of inheritance part 1 was done a little long ago. So it is good that if you ask your friends question, you will also have a revision. Okay, let's do it in every class of mine. Yes, audible, visible. Yes, I saw that, Trisha. I'm so happy that you guys ask questions to each other. Okay, very nice. Yes, those who joined now, quickly mark your attendance and share the link with your friends and let them also join. And also let me tell in the Telegram channel as well, because some of them would have waited. Okay, some of them would have waited and ran away. Okay, yes, visible, audible, perfect, perfect. Yes, so many of you were requesting me for, part 2. So I was like, okay, I'm coming, guys. 1 second. Just give me a minute. I will just inform on Telegram. Yes, done. Yes, awesome. Very nice, very nice. Oh, Trisha has become our new attendance person. Okay, so students, last class what did we do? We did up to replication. I gave you a small example of semi-conservative replication. I mean, I told you that this class I'll be explaining you that, right? So let's start. Okay, voice is not visible. That is very sad. Yeah, very sad. Okay, let's see what is given in NCERT. So for those students who do not know, we are doing NCERT line by line. That means I've taken the snips from NCERT and I'm going to explain NCERT and also make you understand the chapter. Okay, yes, very nice. Now let's see replication. While proposing the double helical structure for DNA, Watson and Crick immediately had a scheme for replication. So students, if they ask you who are the people who ever brought up this idea or ever spoke about replication, what will all of your answers be? Watson and Crick. Okay, Watson and Crick you have to respect enough because they have come up with many things. Watson and Crick gave us the double helical structure. Again, Watson and Crick is the people who spoke about replication first, and they tried to prove it in prokaryotes as well with the experiment. Again, Messelson and Stahl proved it. Watson and Crick spoke about the replication. Clear so far? Okay, I started, you people didn't start, huh? I started already. Okay, I started teaching. Now come back and join. So to quote their original statement that as it is, okay, what is the statement that they told? It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic engineering. I know all of us, none of us are Shakespeare, so we will not understand the statement. Here, what are they talking about, students? They're telling that the possible pairing, that means A binding with T at Gopika Ma'am's class, G binding with C. This pairing that they gave, that they spoke about in their double helical structure, later Chargaff gave a rule called Chargaff rule, right? These things, they were like, okay, there is something about this pairing. It's not just a simple pairing. There's something about this pairing that has some connection with replication. That means DNA has to make their copies. If it has to go somewhere, it has to make the copies. Now, how do they make the copies? Yes, so far good. Thumbs up in the chat. I want energy in the chat. I cannot see any energy in the chat. Students, give energy. 40 marks chapter. Okay, 40 marks chapter. So when did they say this? Watson and Crick quoted this statement in 1953. The scheme suggested that the two strands would separate and act as a template, okay, for the synthesis of the new complementary strands. So here what are they talking about? As I spoke last time about parents and sibling example, I think all of you are clear about it. So students, let's say this is our DNA. Okay, this is the DNA. This is the DNA. Now what is going to happen? Let's say this is the old strand, okay, old strand. These are going to act like templates and form the new strand. Okay, act as template and form the new strand. So this becomes the old strand. this one becomes the old strand, and this becomes the new strand. Now when I'm speaking about the old strand and new strand, what do you understand? The old strand is the template that I took, right? The template I took. I'm giving you a Xerox copy. I'm telling you, go take Xerox and come. What will you tell? The original copy that I give you becomes the template. The new copy that you took from the Xerox machine becomes the new strand or the daughter DNA. Understood? Now, so they are telling that if you take a scheme where you can separate the DNA strand and tell that one DNA strand, you are going to be the template. Okay, that is what is going to happen. Now, look here, after the completion of the replication, each DNA molecule would have one parental, one newly synthesized DNA. Is there any student who hasn't understood the statement from NCERT? Again, listen to me carefully. Each DNA molecule, look at that molecule. Look at the molecule I drew there. Each DNA molecule would have one parental. Okay, yes, do they have one parental? The old one is parental, okay, parental, and one newly synthesized strand. Yes, ma'am, absolutely right. Right, in semi-conservative replication, okay, what is going to happen? Xerox shop is closed, so you people will not form copies, okay, replication gone. Okay, so here what is happening, students? Each DNA molecule would have one parental, one newly synthesized. One parental, one newly synthesized, okay. And this is termed as semi-conservative DNA. Clear? Last time itself I gave you an example, so here I am just doing a revision. Okay, I am just doing a revision. Now, the experiment is very important for you. Many questions can come from this. Now, since you are not a board student, you do not have to write story, right? You do not have to by heart the whole thing, but you have to know each and every words used there, which are the, substances used there, what is the medium, how much, in 20 minutes, how much replication is happening. That is your answer. Okay, that is going to be the questions that is going to come. So, listen to me carefully. They grew, what they took, students? E. coli. They took E. coli in a medium containing N15NH4Cl. Okay, that is N15 is a heavy isotope of nitrogen, not a radioisotope. Okay, many students when they are writing, because we studied Hershey and Chase, no, ma'am, everything is radioisotope. Okay, everything is radioisotope, like that. So, listen here. Yeah, chats are not moving because you people are not energetic. Okay, morning all sleepy people woke up and came off. Now look here, students, do not write that they are radioisotope. Okay, they are not radioisotope. Clear to all of you? Radioisotope is different. Radioactive. Okay, they are radioactive. That is different, and this is different. Okay, look here, as the only nitrogen source for many generation. So I'm erasing this. This N15, okay, N15 is a heavy isotope of nitrogen. So this, take this as the parent. Okay, take this as the parent, because we need to prove that if it is going to replicate in a semi-conservative manner, right? Semi-conservative manner. Okay. Now, as the only nitrogen source for many generations, the result was that N15 was incorporated into newly synthesized DNA. Okay, as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient. Okay, this is, students, this is used. Why? So that we can do the separation properly. Now, as a NEET student, is question going to come, oh, which paragraph, which line did they speak about this? No, they are going to directly ask you, where was cesium chloride used, or some PYQ related on that, or centrifugation was done with dash medium. What is your answer, all of you in the chat? Yes, all of you in the chat. So many kanjus in one class comes, this is what happens. All of you mark your attendance quickly, like the video. Quickly. Yeah, eat 5-star and don't do anything. Now answer this. Okay. Otherwise on May 4th, I will eat 5-star and I will scold you people. Yes, very good, Roshni. Ma'am, it is cesium chloride, which we used it for centrifugation to get an idea, right? N15 was incorporated or not. Okay, N15 was incorporated or not. Now, please note that, okay, please note that N15 is not a radioactive isotope. That's what I told you there. It is not a radioactive isotope. Clear or not? Okay. And it can be separated from N14 only based on the density. One is heavy, one is light. What will you do? You will just do centrifugation, okay? You will just do centrifugation, and you will understand that the heavier one will settle in the bottom, the lighter one will go into the supernatant. So you used cesium chloride, you did the density gradient over, okay? Now then, so students, first step is clear. They took N15 medium and they incorporated. They noticed that the newly synthesized one, both of the strands were N15, N15. Okay, both the strands were, this is the DNA, both were N15, N15. All of you tell me in the charts, why did we take E. coli? Why did we take E. coli? Yes. Okay ma'am, why did we take E. coli? Why didn't we not take any other, you know, any other bacteria, any other organism, why, anything, nothing else? Why did we not take? Why did we only stick to E. coli? I have told this answer many times and this question has come many times, okay? So answer that by now. Now see, then they transfer the cells into a medium with normal NH4Cl14 and took a sample at various time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helix. The various samples were separated independently on cesium chloride gradient to measure the densities of the DNA figure. Very, very, very good, because it replicates faster. In 20 minutes we are going to get replication happening, right? 18 minutes, again duplication is there, replication is there, again they are different, okay? That is why that confusion about 18 and 20 minutes is there. Some places we see 18, some places we see 20, but as of now in your NCERT it's given 20, so we will stick to 20. It is because that E. coli replicates faster. Imagine a replication is going to take around 48 hours or 50 minutes, 60 minutes. Do you think we can do the entire experiment in the time that we planned? We cannot do it. So we have to take a take a prokaryotic cell which is going to replicate faster. Clear? Okay. Now students, so what has happened here? Carefully understand the experiment with me. First they took N15, which was the heavier isotope. But is it a radioactive isotope? In the chats, yes or no? Is it a radioactive isotope? So they took N15, and in first N15 they noticed that the entire DNA was N15, N15. That means both the strands were N15. Then they transferred that into a medium which had N14. So first medium was N15, N15H4Cl. Second medium was N14 H4Cl. Okay. Cl. Okay. Now what has happened students, in the second one they noticed, very good, very good, in the second one they noticed that the strand that they got was a hybrid. The strand that they got was what? Hybrid strand. That means one strand was N15, one strand was N14, indicating that DNA in prokaryotes replicate by semi-conservative manner. But that is enough. Okay, it's not visible. One second. Better now? I think it should be better. Just that I'll get more. Yeah, it's better, right? Yeah, it's visible. Yes. Now it's visible? Yeah, it's better now. It's better now. Yeah. Okay, now look here. Students, after 40 minutes, they noticed that. So what you have to do, I'll tell you this. See, listen to me carefully. How you can do this experiment, I'm going to tell you. See, when you're taking the first medium, okay, first medium. Listen carefully, yes ma'am, it's visible clearly, awesome. Okay, students, when you took the first medium was N15, okay, N15H4Cl, correct? Now in the second one, what did you do? You transferred. So here what you got? You got both the strands which are N15. Okay. If you listen to this carefully, you will be able to save your 4 marks, because always one question keeps repeating from replication and it is usually a numerical. Now numerical means what? Like they will ask you after 40 minutes what happened, after 60 minutes what happened, after 80 minutes what happened, after 100 minutes what happened. So you have to be careful. So look here carefully. N4Cl is there. So in the first thing you got both N15, N15. Yes or no? Now I transferred this E. coli into a medium, okay, I transferred this E. coli into a medium which was a normal medium of N14H4Cl, okay. Now what did I observe? I observed that I got one strand of N14 and I got one strand of N15, correct? Yes ma'am, this is what we got, okay, this is what we got. Yes. Now, what is that reflection here? It's a cardboard that is kept in the front, students. Everything is reflecting on the board. Okay. We have to adjust. Can't do anything. Okay. N14, N15. Got it? Now we got a hybrid. Now, since we got a hybrid, okay, since we got a hybrid, then we wait for another 20 minutes. So, this happened in 20 minutes. This happened in 20 minutes. Now, next one we are going to see. Now, imagine you do not know how to do the calculation, okay? You do not know anything about this. I will give you a way to do it. Now, students, it's very simple. What you do is you separate these strands, okay? Let's say you get one strand of this, yes or no? You separated this and you get one strand of this, correct? Now, in both the strands, you are going to put N14 itself. Okay, what are you going to put? N14. That means you get 50% light, 50% hybrid. Is this clear to all of you? Yes or no? So, always remember that you have to separate the strand, and for each strand you are going to give a N14, because we are not going to keep it anymore in N15 medium. We are going to leave it in N14 medium. So, as it continues and continues, the hybrid is going to decrease in number. So, here what has happened? You are going to get 50% light, okay, 50% light, and you are going to get 50% hybrid. Clear? Now, you want to do the next step. How will you do the next step? Simple as that. Let's say two strands are there. I'm going to separate all the strands. I got one yellow. These two strands I'm separating. One more. Okay. One more. And one blue I got. Now, what did I tell you? To all this, I'm going to add N14 itself. So, I'm going to take yellow again and I'm going to draw this. 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 Now, you tell me what's the percentage, all of you in the chats. Hi, Satvik. Tell me, students, what's the percentage? So simple. Look at the percentage. You are getting 3 light, right? This is light, 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 and hybrid. That means you are getting 75% light, right? You are getting 75% light and you are getting 25% hybrid. Is this clear? If you feel that no, I do not know to do the calculation, always do this technique. With this technique, you will never go wrong. Okay, you will never go wrong. Now, if you want to do further, separate all these strands, you are going to get the next one. Yes, very good. 25% hybrid, right? 3:1, 75% light. Clear? Again, when you do further, again you will get maybe somewhere between 87 and 12.5, some that range you are going to get the next one. That is, you separate all the strands and you continue the process. But is this clear? So, students, how are we going to do the whole process? We are going to do with the help of centrifugation. Okay, centrifugation. So, here what are we going to do? We are going to take the technique and we are going to do centrifugation. Okay, we are going to do centrifugation. Okay, centrifugation, and centrifugation is going to help. How centrifugation is helping? Because of the differences in density. Okay, differences in density. Any doubt in this experiment? So, they are going to ask you question in 80 minutes how much E. coli you got, in 100 minutes how much E. coli you got, in 40 minutes how much E. coli you got. You should be in a position to answer that. Okay. So, let's go back here to the to the experiment, and this experiment was conducted by Michelson and Stahl. Okay, Michelson and Stahl. They did not tell that replication happens that way. That was done by Watson and Crick. But the experiment was done to prove in prokaryotes was done by Michelson Michelson and Stahl. Okay, now students, so look here, after 20 minutes they got 2 hybrids. Okay, again when they separate, so how did they get the hybrid? Very simple, they separated these 2 strands, they added N14 to it. Okay, again after 40 minutes what did they do? They separated all these strands and they got 4 strands. In that you can see N14, okay, N14 to 1 light strand, again 1 more light strand and 2 hybrid strand. So this is a ratio of 50 and 50%. Okay, 50%, 50%. Clear? Absolutely clear? Clear? Yes, okay, done. So light is done, dark is done. Now see, thus the DNA that was extracted, okay, thus the DNA that was extracted from the culture 1 generation after the transfer from N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes. So in NCERT 20 minutes is given. Again, see, remember that 18 minutes, 20 minutes I have read in many places. 18 minutes is for duplication and 20 minutes is for replication. Now if we go into the depth it will become too much, but as of NCERT we are going to stick to 20 minutes. Okay, had a hybrid or intermediate density. DNA extracted from the culture after another generation, that is after 40 minutes. Okay, after 40 minutes, second generation was composed of equal amounts of hybrid and equal amounts of light DNA. Will any of you make mistake? Will any of you make mistake? Should I give you a numerical? Yes. Oh my god, so less. Yes. Clear? Okay. Now you will be able to do numerical. Now students, this was done in prokaryotes. Yes or no? This whole experiment was done in a prokaryote, right? Okay, prokaryotes. Now we are going to do another person called Taylor. He did it in eukaryotes. That is, he wanted to check if all the DNA is going to do same semi-conservative replication. So he did the experiment in Vinca-Rossi. Okay, he did the experiment in Vinca-Rossi. So very similar experiment involving uses of radioactive thymidine they had taken. Thymidine they had taken to detect. Now, question to all my students. Thymidine, is it a nucleoside? Is it a nucleotide? All of you in the chats, quickly answered me this. I have told you that there is a suffix for it, and what is the suffix and what it is indicating. All of you open your notes, quickly open your notes. Yes, students, and quickly like the video. Why are you people so confused? Yeah, why are you people so confused? What is there in liking the video? Right, it's a help for that. You're showing that, ma'am, I liked your class, that's why I'm liking. All of you tell me in the chats, is thymidine, radioactive thymidine, is it a nucleoside or a nucleotide? I-D-I-N-E. Let's see who is going to answer. Very good, Madhumati, very good, Charan, very good, Sweetie, very nice. These 3 answered the first, that is a nucleoside. Okay, Mercu, I cannot even pronounce your names. Okay, yes, Sukanya, Minecrafter, Tejaswini, Satvik. Okay, very nice. This is a nucleoside, right? This is a nucleoside and it is a pyrimidine. Now, detect distribution of newly synthesized DNA in chromosomes. So you have to remember just the name. Okay, so they did it in Vicia faba or faba beans by Taylor and his colleagues in 1958. Okay, yes, the experiment proved that DNA in chromosomes also replicates semi-conservatively. Okay, so it was done in faba beans. It's a variety again. This faba beans, it was done in 1958. So do not forget Taylor et al. They tell. Okay, Taylor and his colleague. Taylor and his colleague. Et al. Is actually, students, when we write research paper, if we are not the first author, okay, it's usually the first author's name that is going to come. So Taylor, let's say the first person, the rest everyone came as et al. Okay, everyone will come as et al. Understood? Taylor and his colleagues came up with the experiment to prove that in eukaryotes also, okay, in eukaryotes also DNA is going to replicate in semi-conservative manner. Done, done, done. Is this clear to all of you? Okay. Semi-conservative manner is clear, no? The word itself indicates, see, what does the word indicate? Semi means half, right? Semi means half. Conservative means conserve. Conserve. So that means they are telling that 50% is going to be parent, okay? 50% is going to be parent and 50% is going to be the new strand. That means they are going to conserve 50% and 50 is going to be new strand. From the name itself, it is clear. Moving on, student. Yes, Kohinoor clear. Okay, moving on. The machinery, okay. My throat has a problem again. This keeps on and off. Yes, let's go. Yes. So students, here let's see the machinery. That means, see, now you learned replication, but did you learn how both the strands are separating? Did I tell you anywhere that how both the strands are separating? For that we have a set of enzymes that is going to do the job. So let's see which are the enzymes. In living cells such as E. coli, the process of replication requires a set of catalysts or your enzymes. So in your NCERT, they have given very few enzymes, but you need to know some more enzymes to understand the process completely, which I'll be teaching you. Okay. Now, the main enzyme is referred to as DNA dependent DNA polymerase. That means after I separate the strand, I need one person to make new strands. Yes or no? That person who's going to form a new strand for me or new template is going to be my DNA polymerase, that is DNA dependent DNA polymerase. Now, since it uses a DNA template to catalyze the polymerization of deoxynucleotides, these enzymes are highly efficient enzymes as they have catalyzed polymerization of a large number of nucleotides in a very short time. So, DNA polymerase is a very efficient person. Why? Because in a very shorter period of time, he'll get more work done. Okay, more nucleotide addition is going to happen and he's going to form the new template. Okay, now look here, students. E. coli has only 4.6 into 10 to the power 6 base pairs compared with human. Human has how much in the diploid content? 6.6 into 10 to the power 9 base pairs. Completes the process of replication within 18 minutes. So here is where the confusion is. So look here, it with human whose diploid content is 6.6 into 10 to the power 9 base pair completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pair per second. Understood? If they add 2000 base pair per second, then it is going to happen. The replication is going to get over in 18 to 20 minutes. Otherwise, it is going to be a slower process. So, DNA polymerase is very efficient. Why? Because it can do the job in a shorter period of time. Understood? Got it? Now, not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. You tell me, if one nucleotide changes that side, this side, what is going to happen? All of you in the chats. So many questions I'm asking people today. Students, let's say, okay, let's say G became C. Okay, G became C. What is going to happen? In the chats, all of you know the answer for this. Quickly. Quickly, all of you, tell me this. Tell the answer for this faster. I think, yes, very good. Mutation is going to happen. Yes or no? Mutation is going to happen, students. So, if the DNA polymerase does not form the new template absolutely with high accuracy, anything can go wrong. Any mutation can happen, right? So, look here. Any mistake during replication would result into mutation. The answer was here. Still, you people took so much time. 2 days you took. Okay. Furthermore, energetically, replication is a very expensive process. Deoxyribonucleotide triphosphate. Okay. Deoxyribonucleotide triphosphate. Now, I have taught you this. All of you go to my first class and all of you remember when I wrote it, I told you that a sugar, okay, a sugar plus what is going to form your nucleoside? Sugar plus base is going to form nucleoside, correct? This is a nucleoside. Can you all see if I write it here? Now students, to this nucleoside, if I add a P, can I call this nucleoside monophosphate? Will you all agree to it? Monophosphate. Nucleoside monophosphate. If I add one more P to this, I will call it diphosphate. Triphosphate. So here what are they talking about? Deoxyribonucleoside triphosphate. Right? Understood how this phosphate came? So this triphosphate is when we add 3P to that S in B. With the nucleoside, if you add 3-phosphate, we are going to get deoxynucleoside triphosphate, which serves as a dual purposes. This last class I have drawn a diagram beautifully and explained all of you, okay? So I hope all of you are remembering. This is a revision class because this chapter we have previously done 2 times, okay? Now, in addition to acting as a substrate, they provide energy for polymerization reaction. The 2 terminal phosphates in the deoxynucleotide triphosphates are high energy phosphates, same as in case of ATP. So for those students who did not understand, raise your hands. Raise your hands. So what are they telling students? Yes, very nice. So here what is happening? So GM Express we are going to do. See students, S plus B. Oh, two people are writing. Okay, S plus B, that is sugar plus base. Okay, let me just write that only. Otherwise students will be like, what has ma'am written? Sugar plus base will form what? Ma'am, obviously it will form nucleoside. Right? It is forming nucleoside. Correct? Agreed? Now I am going to add phosphate to it. Okay, now I am going to add phosphate to it. So when I add my first phosphate, okay, when I add my first phosphate, it is going to, if you understand this only, you are going to understand everything further. So listen to me carefully, don't be like, hey, this is not going to come as a question. It will come as a question, okay? So students, this whole thing I will call it as nucleoside monophosphate. Correct? Now if I add one more phosphate to it, okay, if I add, let's say 2 more phosphate, I'll directly go into triphosphate. Okay, phosphate, 1 more phosphate. Okay, what is going to happen? I can call this whole combination from here up to here as nucleoside triphosphate. I hope this is clear to all of you. Who is the energy production? Okay. Energy production, yes. So this energy production, nucleoside triphosphate. Now how is it deoxynucleotide triphosphate? If the sugar molecule is a deoxyribose, yes or no? If the sugar molecule is a deoxyribose, that means in the second position we have a H instead of an OH, it is going to be deoxy. So deoxy triphosphate is going to be the energy yielding process in your entire machinery or formation of your replication. Now they have mentioned one more important statement telling that these 2 phosphates, when they are breaking, okay, when they are breaking and moving, okay, that is going to, okay, GM Express is Gopika Ma'am Express, where I teach you everything faster, okay, like I give you an overview of what we just learned, okay. This is going to happen during your NCERT line by line, because we'll read NCERT and then I'll give you a quick recap like this. So students, they are telling that when we break these 2 phosphates, okay, when we break these 2 phosphates, they are going to be high yielding, okay, high yielding of energy. Got it? Just like your ATP, these people are going to break and they are going to yield high amount of energy, and this energy is going to help in polymerization. Every line of NCERT is clear. Like this, we are going to learn the entire chapter where I will read NCERT, then again I will explain you. Done, done? Perfect. Yes, take the ticket and then only enter GM Express, or else someone will be there to check. Rathas will check your tickets. Done. So this portion of the statements are clear. Now the next statement that has come is that these machineries that we are using, machineries means like the enzymes that we are using. Which are the enzymes? So students, look at this. This is my DNA strand. Okay, this is my DNA strand. Now this DNA strand, as you can see, there is a double helical structure. They are very wound to each other. Now I have to remove them, yes or no? I have to make sure that I have to get a template out of it. So when you are learning this chapter, I want you to connect everything to the previous topic. Don't sit like this and be like, huh, why should I unzip this? Because you forgot about replication. Don't be like that. You should connect all the topics. Clear? Yes. So here what happens, students, your two strands are there. Now if I have to unzip it, I am going to take the help of an enzyme. Who can tell me the enzyme? In the chats, who can tell me the enzymes? See, I started unzipping from one end. So the enzyme who is going to unzip is going to be none other than, you people will tell. Yeah, very nice. Stay outside only. Tell me quickly the answer. Yes. Here what is the unzipping enzyme? The unzipping enzyme is going to be your helicase. Okay. All of it you people forgot. Students, without revision, I am telling you, don't think biology is easy and you will put all your concentration on physics and chemistry in the last minute and you will forget every single word of biology. Biology, you have to revise every then and now. See, before this class also, I went through the whole NCERT, because even if I am a teacher who has taught it 3-4 times, even I tend to forget terminology. So that means every person has to revise. That is how our brain works. Okay, every teacher goes through the notes before entering into the class. Be it however big of a teacher, they will do that. Why? Because every human has that tendency. We have so many unnecessary information that we might sometimes forget the necessary information. Understood? You have to revise. Helicase. So helicase is also called as the unwinding enzyme. Okay, why is it called unwinding? Because it is unwinding. Wow. Okay, what an answer. Okay, because it is unwinding, right? The strand which was like this, I unwound into this. So helicase is going to unlock like this. The DNA strand was like this, it started doing this. Okay, it will start like this and it was going to separate into two strands. Got it? Yes. So here you can see the strands which are started completely unzipping like this. Okay, and this opening that you're seeing looks like a fork. So this is called as your replication fork. What is this called as? Your replication fork. Clear? So far good. Can we go back to NCERT? I just gave you an idea for this. Okay, I just gave you an idea. Now we are going to go back to NCERT and see what NCERT is telling. Now till this last statement, I have explained every single thing to you. This is all general, but you understood that DNA polymerase will form the new strand and it has to be highly accurate. It cannot make a mistake. If it makes a mistake, that is a mutation. Okay, that is a mutation. Very nice. Okay. Yes. Now students, look here. In addition to DNA dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length, because understand, these both strands are super coiling, right? They can come back very easily to each other because they are complementary. They can come back to each other. Base pairing is there. So we cannot unzip the whole strand and be like, hey, this strand is the template, this strand is the coding strand, throw them both the sides. You cannot do that. You have to make sure that you're unzipping small portions and their DNA polymerase is forming new strands, new templates and adding on. Okay, because they have a tendency to supercoil. They have a tendency to re-anneal. For that also we are going to use different enzymes. Clear? Okay. Now look here, students. Clear so far? Thumbs up in the chat if it's clear. Because I'm getting scared looking at your responses. Is it clear to all of you? Some people are answering, some people are dreaming. This chapter, I understand, it can get a little complicated, but trust me, it's not that complicated. Okay, it's not that complicated, just that it's a lot of content. But since I'm reading the content for you, it should be easy for you now. Okay, now look here. DNA cannot be separated in its entire length due to the high energy requirement. The replication occurs within a small opening of DNA helix, which is referred to as replication fork. Is this clear why I told you that is a replication fork? Because the mouth is opening, right? Same way like this, the strands are opening. Here they will start adding the new template. The entire strand cannot be unzipped. Okay, now the DNA dependent DNA polymerase catalyzes polymerization only in one direction. This is one of the conditions, very important conditions. Okay, I'm going to mark it with a different color, and all this PDF you will get it on Vedantu NEET English Telegram and on my Telegram, that is Biology by Gopika. So join the Telegram and you will get the notes. Yesterday's notes also I have uploaded there. So look here, not yesterday's, Tejas notes. See, the DNA dependent DNA polymerase catalyzes polymerization only in one direction, that is the 5' to 3' direction. Okay, 5' to 3' direction. Okay, DNA batch is available now with Tejas series. The entire batch is free, so I would request you to go for that, because you will get the book also, you will get the batch also, and all the recorded lectures are already in the batch, and we have only taught the lectures, all the important chapters we have taught there. So again we have one more month of, we'll finish the portions. So all the recorded lectures are there, VDPPs are there, books are there, test series will start. Okay, all India test series will start once we complete the portions. So I would tell you, get both of it together. Okay, because individually if you're going to pay, it's going to be more. So when you buy together, it will be better. Okay, so get Tejas plus this, and Tejas also has a lot of practice questions, so you can practice a lot. Now students, look here, this creates some additional complication at replication fork. Consequently, on one strand, the template with polarity 3' to 5', the replication is continuous, while the other template with the polarity 5' to 3', it is discontinuous. The discontinuous synthesized fragments are later joined by DNA ligase. Now just reading this, I know all of you will not understand most of it, so we will go further again in detail. Okay, but this paragraph is very important. Many times I've seen question from this S-phase question. Okay, not available. Wait, I will check and tell you. I will drop the link in the Telegram. Okay, furthermore, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at the S phase of the cell cycle, right? You have phases of the cell cycle. All of you tell me one phases of your cell cycle. Yes. Right, you have G1 phase, right? S phase, G2 phase, you have interphase, the entire whole phase. Okay, interphase. So here what are they telling? DNA takes place at the S phase of the cell cycle. The replication of DNA and cell cycle division should be highly coordinated. A failure in the cell division after DNA replication results in polyploidy, a chromosomal anomaly which you have studied in principles of inheritance. Yes or no? You will learn the detailed nature of origin and process occurring at your higher classes. That means in MBBS you will be studying it. Yes, G1 S phase and G2 phase, and you have a G0, 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 many terms are there for that, where it is a complete resting phase, right? So here what is going to happen? DNA replication is going to take place at the S phase, and they are telling that if cell division does not happen properly after the replication, it will result in polyploidy. Now we will come back to replication. Okay, we'll come back to replication. All of you are ready? Okay, look here students. So you understood from where energy is coming, you understood from where your strands are coming. Now I will teach you some more enzymes that is important as a NEET student. Okay, now for boards we will not teach this much, but as a NEET student you should know little more additional information. Look here students, your normal strand, we are going to form the strands. Okay, we are going to form the strand. Oh no, yes. Why can't I get a copy of this? Okay, never mind. Okay, look here students, this is your replication fork that I am drawing. Okay, this is how your DNA strand is going to look. It looks fine, no? Yeah. Okay, now look here, we already understood that helicase is the one who is going to unwind. Okay, so we are going to write it here. Helicase is the one who was unwinding. So helicase is continuously doing the job. So now students, as I told you, they have a tendency. Who has the tendency? These two DNA strands have a tendency to re-anneal, come back together. So for that we have another enzyme that is going to help, actually like a protein. Okay, again enzymes, proteins all are same. There is one more person which is called as your SSBP. Who can tell me the full form of SSBP in the chat quickly? So students, here let's say we have another enzyme which is called as your SSBP. What is SSBP full form? I've taught you this also last time. SSBP, which is single strand binding protein. Clear? Yes? No one told? All of you forgot, huh? Single stranded binding protein. Very good, Satvik. Ma'am, it is single stranded binding protein. Now, what is this protein going to do? Remember that this protein is going to prevent or avoid re-annealing of both the strands. Okay, re-annealing of both the strands. That means they have a tendency to come back together. That will be stopped by your SSBP. Okay, fine, understood, ma'am. Now, DNA polymerase students have to do the job. What is DNA polymerase job? To form the new strand. But DNA polymerase has many conditions. Okay, he's the hero, no? He has many conditions. That I will not work in hot, I will not work in this set, I will not work with this director. Like that, you know, big heroes will have so many conditions. Same way, this DNA polymerase also has so many conditions. The first ever condition in DNA polymerase is that, okay, DNA dependent DNA polymerase, I'm not telling over and over that. Okay, DNA polymerase is that first condition is that I will work only on a single strand. What is that? I will work on a strand which is a single strand. That means both the strands have to be separated and I will work on a strand which is a single strand. Second condition is I will only work on a strand whose polarity is from 5' to 3'. Clear? I will work on a strand whose polarity is from 5' to 3'. Clear? Yes? So far good? All of you? Okay, fine. Okay. So students, this is the two condition. Then obviously hero, no, we need hero. Without hero, movie will not work. So we were like, okay, fine, you come. Okay, now the DNA polymerase is ready to start working. But DNA polymerase has a problem. It does not know exactly where to start the replication. DNA polymerase does not know where to start the replication. So then we'll be like, okay, fine, whatever. Our faith is bad. So we will have to bring one more person, and that is going to be a primer. Who is that person? Primer. This primer, students, we are going to add these primers in the starting. Let's say now, since I drew SSB here, I'm going to add the primer here, and this primer will tell that, okay, DNA polymerase, you know what, you should start the replication from here. Before that, let's label the strand so it's clear to us. 3' to, so 5' to 3'. Okay. Now students, the DNA polymerase will only work on a strand from 5' to 3'. So I added the primer. Now DNA polymerase will be like, okay, whatsoever, I will start continuing the formation of the template. Okay. So let's say the template formation is going to continue. Got it? Now here he formed a new daughter DNA which is from 3' to 5'. Got it? Clear? Now, there is a problem again, because students, we are not going to unwind the entire DNA strand together. There is chances of supercoiling. Okay? Yeah, villain will come. Okay, villain will. Hey, you people, without cameraman, without villain, tell, without heroine, without, you know, tea, coffee, then grasses, everything you tell. Okay, for everyone I'll give a role here. Only 5-6 people are there. Now students, because the DNA we are not unwinding together, there is a chances of supercoiling. Okay, there are chances of supercoiling. So here at this point, to avoid supercoiling, let's say, now I actually have to draw it further. So I'm just drawing it here. Okay, I'm just going to draw it here, and I'll extend the strand. To avoid supercoiling, we are going to use topoisomerase. Okay, we are going to use topoisomerase. This one is topoisomerase, who is going to protect from supercoiling. Okay, it's going to prevent supercoiling. Clear? Supercoiling will be prevented by topoisomerase. Okay. Now, listen carefully. Next problem that we have, all of you tell me what is the next problem that we have. The biggest problem is DNA polymerase will only work on a strand from 5' to 3'. The next strand that we have is from 3' to 5'. Now how do we make DNA polymerase? How do we convince this hero to work with this director? For that, primer will be like, you know what, I will do one thing, I will start adding it reverse. Okay, I will start forming it from the reverse. So we will add the primer in this end, 5' end. Okay, like this we'll add the primer. Okay, then DNA polymerase will not get to know. DNA polymerase will start forming new strands. Again, because it's in the reverse, more energy will be required. Here again primer addition will happen. Okay, again the DNA polymerase will continue. Okay, so this will keep happening, students, for some time. Okay, like this, and again DNA polymerase will continue forming the template, and the form template is going to be 3' to 5'. Okay, like this. Got it? Clear? Now after the job is done, everyone is the same, right? Every human is the same after the job is done. So after DNA polymerase has formed the new strand, DNA polymerase is like, why do we need the primer? Remove the primer. Okay, so now we will be forced to remove the primer. That means we will remove that director and throw the director away, telling that we don't need any more script, I don't need anyone to lead me, I am good this way only. So we will remove the primer, and to make sure the spaces where the primer it was there, no, that is going to form fragments, no, because look here, in between primers are there. 1 second, students, this erasing is going to be a task. Okay, like here the primers are there, which is going to lead to some spaces. There the nucleotides will be formed by DNA polymerase itself, but the gluing will be done by ligase. Okay, gluing will be done by ligase. So during the gluing, we will use an enzyme which is called the gluing enzyme, which is called the DNA ligase. Clear to all of you? Like a story, you remember this, don't by heart anything. Okay, everything is a story. DNA ligase will do the job of gluing. Now students, these fragments which we got, what is these fragments called? These fragments are called 𝐂𝐡𝐞𝐤 fragments, after the scientist name. Okay, not just scientist, the person who figured this out. So these fragments are called as 𝐂𝐡𝐞𝐤 fragments. Okay, 𝐂𝐡𝐞𝐤 fragments. Because they are formed as fragments, yes or no? The fragments are again joined or glued together with the help of DNA ligase, but the nucleotide formation is happening again from polymerase. Okay, ligase do not have the do not have the ability to form the nucleotides then glue it. Okay, that job it will not do. It will just glue, sticking. Only what will gum do? You tore a paper, will gum stick it, like fix all the pieces of the paper and then glue it for you? No. If you put glue, the paper will get stuck, right? So 𝐂𝐡𝐞𝐤 fragments, and as you can see here, this strand which is formed on the 3' to 5' end is a discontinuous strand. Yes or no? It is a discontinuous strand. So we will call this strand, as discontinuous or lagging strand. Discontinuous or lagging strand. Okay. And this strand which is continuous, in one shot we did this, no, we did not have any problem, everything was fine. This strand we will call it as continuous, okay, continuous or leading strand. Clear to all of you? Is the whole process clear? How many enzymes did I teach you? I taught you helicase, I taught you SSBP, I taught you DNA polymerase, primer, DNA ligase, topoisomerase. Around 6 enzymes are there, but only 2 enzymes are mentioned in your NCERT, but it is important for you to learn all of it. Okay, very good, very nice. Okay, what are you guys telling? NRG. Okay, leading strand. So the whole DNA polymerase replication is clear. Any doubt? Should I write any points anywhere? Tell me in the chat. Okay, now there is one important thing, that is, students, if the replication has to start, there should be a spot where replication has to start. See, primer is going to lead the strand, that is a different story, but there should be a place, there is an exact location at which the replication will start. That point is called as ORI, that means origin of replication. Okay, origin of replication. Clear? Origin of replication is the place or the exact site at which the replication is going to happen, and this we have learned when we learned vectors, that they should have an ORI site. Yes or no? So ORI site is the region where the replication is going to start. So ORI, you have to remember that this is called the replication fork. Okay, this you have to remember that this is called the replication fork, the portion which is unwound and where the new strand will start forming. Okay, new strand will start forming, and this is again semi-conservative replication. No, then students, you have to remember continuous leading strand, discontinuous lagging strand. You have to remember the polarity. DNA polymerase will only work on a strand whose polarity is from 5' to 3', and it will work on a strand which is a single strand. Okay, primer is the person who is going to lead the DNA polymerase or tell that this is where you have to start the replication. Okay, then SSBP is a single stranded binding protein which is going to avoid re-annealing of both the strands. Topoisomerase is going to avoid the supercoiling. DNA ligase is going to do the gluing of the enzyme. Easy peasy, clear? Yes, all of you? Super easy, no? Okay, fine. Very nice. Okay, this is done. So students, here there is one Kuti diagram given in your NCERT. All of you can see the diagram and you can see how they are. See, 5' to 3', it is going to be a continuous strand because at one shot we formed it. Okay, 𝐂𝐤 right? Here you see from 3' to 5', it is a discontinuous strand, and each of these fragments are called as Okazaki fragments, and they will be glued with the help of DNA ligase. Okay, now template DNA strand, again template, one of it is going to be the template, the next is going to be the new strand. Okay, yes. Then this is also clear, no? DNA replication takes place at the S phase, and in case replication does not happen in the correct time, then it is, I mean, cell division and replication are not like going hand in hand, then it is going to lead to ploidy. Okay, polyploidy. Done. Why are you spoiling my name, Sony? All of him call him Pony. S you change and put P. Okay, yes. Students, next thing. Students, now all of you, I hope, remember central dogma. Yes or no? All of you remember central dogma? Central dogma. What is central dogma? Ma'am, very simple, ma'am. DNA will become RNA. RNA will become protein. This is central dogma, right? Now, remember that DNA forms its own copies. We call it replication. We just now learned that replication is going to happen by semi-conservative manner. Right, semi-conservative manner. Okay, and the process where DNA becomes RNA, the process where DNA becomes why they should stop? You did not stop. No, I told you from last class, you did not stop only. Sony is feeling bad, guys, so we will stop. Okay. She's putting emojis. Okay, she's putting emojis. I cannot put any emoji, no, that's why. Yes, okay, this is all for fun. Okay, fun, but don't take it seriously and start calling every day. Okay, yes, all of us have a beautiful name, so let's use our beautiful names. DNA and RNA. So students, DNA to RNA is going to be called as transcription. Okay, RNA to protein is going to be called as translation. And this protein, how are they formed? They are formed by many amino acids combining with the help of a peptide bond. They will form the polypeptide chain. Yes, polypeptide chain. Clear? Yes or no? Yes, clear. Okay, all of you clear? Okay, done. This whole thing is clear, central dogma. Now we are going to learn transcription. Okay, now we are going to learn transcription. Listen to me carefully. Transcription is very easy. Only few terms you have to remember, few things you have to remember, like points you have to remember, that I will teach you. Now look here. The process of copying genetic information from one strand of DNA into RNA is termed as transcription. Okay, it's termed as transcription. Here also the principle of complementary governs the process of transcription, except the adenosine complements now forms base pair with uracil. So students, here what is the only replacement that is going to happen? Here the replacement is going to be that adenine is going to bind with uracil instead of thymine, because we are going to form mRNA. So sorry, we are going to form mRNA and we are not going to form DNA. So since we are going to form RNA, what is going to happen? Thymine will be replaced by uracil. Is this clear? Okay, this is the statement given here. Unlike in the process of replication, which once sets in, the total DNA of an organism gets duplicated. In transcription, only a segment of DNA and only one strand is copied into RNA, which is a very, very important thing. I have explained to you last time also why only one strand is going to form RNA, why not both the strands. Okay, why only one strand? Okay, is going to form RNA, why not both the strands? So again we are going to explain this. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. Now, why both the strands are not transcribed? Okay, why both the strands are not going to be copied during transcription has a simple answer. Listen to this carefully, and you have to answer it. Okay, students, can I get some hot water? Will you all wait for 1 minute? Okay, just 1 minute. Just give me 1 minute. I'll just get some hot water. I don't know from where cough came. Yes. Okay, look here. I'm back. Okay. So students, here they are going to explain why we are taking only one DNA strand to form RNA. Why not we are taking both the strands? DNA is a double helical structure, both the strands are complementary to each other. Then they'll be thinking, why can't we take both the strands? So look at the answer here. Okay, first, if both the strands, okay, if both the strand act as a template, they would code for RNA molecule with different sequence. Okay, with different sequence. Remember, complementary does not mean identical. Yes or no? Now if I am forming this strand, okay, this is one strand of DNA. Let's say this strand has A, G, C, T, A. Okay, the RNA that this people is going to form is going to be like this. It is going to be U, G will code for C, G, okay, A, U, U, right? This is one strand. Now let's say I have taken the complementary of this one. If I take the complementary of that one, what is going to happen, students? A has code for T, C, G, again what is the T, no? A, T, T. This is the second strand. Now if this strand forms a RNA, look at how it is going to be. It is going to be A, G, C, U, A. Now you tell me, what is the big difference? These two strands are going to be complementary. Both the RNA which I formed is going to be complementary. Yes or no? Let's say this is strand 2. Okay, this is RNA 2. Okay, this is strand 1 and this is going to be RNA 1. Now look at RNA 1 and RNA 2, students, they both are going to be complementary. Strand 1, strand 2 also are complementary, but complementary does not mean identical. That means they have a chance that they will bind back to each other. Okay, so look here that, and in turn if they code for proteins, the sequence of amino acid in the proteins would be different. That is also true. They are not identical, but they are complementary. So when they are going to code, they are going to code this way, right? A, G, C. Coding is going to be different. Okay, here what is the coding going to be? U, C, G. So they are going to code for different amino acids. Now, according to our central dogma, the rule is that one DNA strand will code for one mRNA, and that one mRNA is going to code for proteins. Yes, we do not have a polycistronic gene, we will have a monocistronic one, right? That means this one rule is going to continue, which is going to form a monocistronic gene. Monocistronic gene. That means 1 say 1, okay, 1 to 1. Understood? We are not going to be like prokaryotes where we have one DNA strand which will code for many proteins. Protein. This is a polycistronic gene. Okay, this is a polycistronic gene. I'll teach you about polycistronic and monocistronic again, but just understand poly and mono. Mono means one, poly means many. This is in prokaryotes. We cannot do this. We cannot do this in ours. Clear? Yes? Now, here students, look here. Okay, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, okay, would be complementary to each other. Yes or no? R2 and R1 is complementary to each other. Yes, agreed, or all of you? Agreed, all of you? Okay, now hence would form a double-stranded RNA. Imagine we form a double-stranded RNA. They both got complementary to each other. Will protein synthesis happen? Will protein synthesis happen? Yes? Possible? Protein synthesis is possible? So it will not be possible. This would prevent the RNA from being translated into protein, and exercise of transcription would be futile one. Okay, clear to all of you? So why we are not taking both the strands? Because both the strands will lead to the formation of RNA complementary strand. That means both of them will become double-stranded RNA. Double-stranded RNA cannot get translated. The first thing is, if even though they are complementary to each other, they still have different sequence. So they will start coding for different proteins. Clear? Yes? Very good, okay? Yes. Students, before my voice crack, let's try to learn as much as possible. Okay, then I'll take a break because my voice is cracking. I don't know how long I'll be able to teach, but let's try. Okay, let's try. Okay, don't worry, I'll be fine. Okay, because the weather is so bad here. I think all of you would have seen, no, the flooding and everything happening here. Before my voice crack, we will do. Okay. No, this is not Tejas series, this is NCERT line by line. Okay, Tejas series will only be happening in the evening, so most students can join and it will be helpful for them. Now students, transcriptional unit. So this is a very important question. Transcriptional unit starts with a promoter. Okay, promoter. Structural gene and terminate. This is the unit. Okay, I am not talking about the process, I am talking about the unit. These three people are going to form the transcriptional unit. Okay, now look here all of you. Now students, remember the entire DNA is how much meters? Ma'am, it is around 2.2 meters long. Right, 2.2 meters long DNA we have. Do you think the entire DNA has to become RNA? Do you agree or not? Do the entire DNA have to become RNA? Okay, yes. What happened, Tanuja? Yes, the entire DNA does not have to become RNA. So a small region, okay, we call it a small region of the DNA is going to become the RNA, and that region is going to be called your structural gene region. What is it going to be called? Your structural gene region. Clear to all of you? Yes or no? Clear? Okay, how much are we on the target? Oh. Okay, fine. The entire DNA does not have to become RNA. So we will take a small region of the DNA. That region is going to be called the structural gene. Now the process, students, the process, how this is going to happen, remember, promoter, obviously it is going to help in the initiation of the reaction. Okay, this is the process and unit is different. If you mix it up, okay, you will guys will get nice beatings. Okay, you cannot mix it up. This is the process, this is the unit. Okay, so initiation means where the promoter means what? Something that is going to promote the reaction. So there initiation is going to happen. Structural gene region is where your elongation is going to happen. That means your formation of a new mRNA is going to take place. Okay, then terminator region, obviously termination will happen. Termination is going to happen. Done. All of you clear? Process is clear, unit is clear. Now, students, remember, in each of these processes, one hero-heroine entry is there. See, first, when initiation is going to happen, we are going to take the help of a sigma factor. What factor? Sigma factor. Clear? Now, when termination has to happen, we are going to take the help of rho factor. Now, these, ma'am, is this factors going to actually do the elongation and form the new mRNA? The answer is no. The process or the person who is going to help in the whole formation of all of this is going to be DNA dependent, okay, DNA. Okay, look at with what I am writing.
Oh, okay. Oh, wow. Where is the other one?
Gone. Yeah. Yes, DNA dependent. Yeah, DNA dependent RNA polymerase. Clear to all of you? DNA dependent RNA polymerase. That means this is going to help us in what? This is going to help us in the formation of the new mRNA. Why have I written DNA dependent RNA polymerase? Because it is going to be on the DNA, right? From the DNA it's forming mRNA. So it has to be DNA dependent RNA polymerase. Got it? Yes. Yes. Yes, so it's Mithun sir, okay, he came to check if the studio was fine because morning pen was missing. So now I started writing with this pen, okay. So I was like, time is going, so I'll start writing with this. So he came to check because I had complained, no. Yes, now look here, there is a convention in defining the two strands of DNA in a structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA dependent RNA polymerase, okay, DNA dependent RNA polymerase also catalyzes the polymerization in only one direction. Again students, just like DNA polymerase, this RNA polymerase also has some conditions that I am going to work on a strand that is from 5' to 3'. Does that mean that our problem is solved? How? What do you guys think? Don't you think the problem is solved? Guys, you're all not liking the video and I'm very disappointed in that. All of you are attending, all of you are learning a lot. I want all of you to like the video as well. Okay. Now students, what is going to happen? Okay. Neelam, please come back to the class. We can ask about lunch later. Students, here one thing is going to be very clear, that I cannot take both the strands of DNA. This is my two strands of DNA. One is from 5' to 3', one is from 3' to 5'. Okay, correct? Agreed all of you? Now, this fellow is telling, who RNA polymerase is telling, I will only work on a strand which has a polarity from 5' to 3'. Now, the first question that we asked, why we are not taking both the strands, is kind of solved. Yes or no? Telling that RNA polymerase will only work on a strand from 5' to 3'. Got it? So here that is also sorted. So look here, the strand that has the polarity 3' to 5' will act as a template. Okay, so here what are they going to tell students? They are telling that also catalyze the polymerization in only one direction, that is 5' to 3'. The strand that has the polarity 3' to 5' will act as a template and also referred to as a template strand. Also referred to as template strand. The other strand which has polarity 5' to 3'. Okay, now there is a small twist here. Okay, there is a small twist here. This RNA polymerase is telling that I will form, okay, so here There's a small word thing. See students, he's telling who? RNA. Okay, let's consider this as a hero. Okay, RNA polymerase is telling I will form a strand from 5' to 3'. So if he wants to form a strand from 5' to 3', he is going to work on 3' to 5'. Clear? Clear? All of you? It is not similar to, see, DNA polymerase also had this condition, but he was like, I will work on a strand whose polarity is from 5' to 3'. He is telling, I will form a strand which is 5' to 3'. So I will work on a strand which is from 3' to 5'. Is this clear? Too confusing? Too confusing, or is it clear? Yes? Clear students? All of you? Please put thumbs up if it is clear. Okay, is it clear? Very easy thing. I will not work on 5' to 3'. I will form 5' to 3'. If I am forming 5' to 3', I will work on 3' to 5'. So this strand becomes the template. This strand becomes the template. This becomes the coding. Okay, this becomes the coding. How the irony is so funny that they have called the strand which is not doing anything as the coding strand. All of you, is it clear? Okay, now look here, and the sequence same as the RNA except thymine will be replaced by uracil. Now strategically this strand which does not code for anything is referring to as, okay, coding strand. Strangely, so as I told you, it's an irony that how coding strand which does not code for anything is called as the coding strand. Okay, it's confusing. What is confusing? Tanuja, listen to me carefully. Listen to me carefully. I am telling, okay, what did DNA polymerase tell? DNA polymerase told I will work, okay, only from 5 o'clock to 3 o'clock. Clear? The answer is clear. I will work from morning 5 to evening 3. Enough. That's all I will work. We were like, okay, fine. That means DNA polymerase will work in that slot. Now RNA polymerase is telling that I will work on a strand, or I will form, okay, 5' to 3'. If it has to form 5' to 3', we have to give him the opposite polarity, that is going to be 3' to 5'. Is this clear? Listen to me carefully. One is working on that polarity, another one is telling I will form this polarity. Do you understand the difference? Working on the polarity, forming the polarity is a different story. That's why the statement might be confusing. See, the other strand which has polarity 5' to 3' and the sequence same as RNA. See, here they are telling 5' to 3. So this line is not very clear. Okay, this is why NCERT can be a little confusing. The line is not very clear. Fine. Now they are telling, see, the strand that has a polarity 3' to 5' will act as a template. Template means what? The strand which is actually going to form the mRNA. Clear? So I have made it clear for you. Now it's clear. Tanuja, it's clear. Jasmina, it's clear. Those who told confusing, is it clear now? Okay. All the reference point while defining a transcription unit is made with coding strand. To explain the point, we have a 2 strands. So students, look here. This is going to be the template strand. Okay. And the down strand is going to be coding strand. So whenever RNA is going to be formed from this strand, it is going to be similar to this strand, just that the T will be replaced. Okay. T will be replaced by U. Correct? Yes? All of you? Is this clear? Any doubts so far? Now we will go to transcription. Okay? Transcription. So students, the promoter, the terminator, flank and the structural gene in a transcription unit, the promoter is said to be located towards the 5' end. Okay? 5' end of the upstream. Okay? Of the structural gene, that means on the upstream. You have to remember the upstream and downstream. The upstream, what is going to be present? Your promoter is going to be present. The reference is made with respect to the polarity of the coding strand. It is a DNA sequence that provides binding site for the RNA polymerase, and it is in the presence of a promoter in a transcriptional unit that also defines the template and the coding strand. By switching its position with the terminator, the definition of coding and template strand could be reversed. All this is not required. I'll tell you whatever is required. The terminator is located towards the 3' end of the downstream of the coding strand. Don't get it confused again telling that, ma'am, you told 3' to 5', now they're telling 5' to 3'. They're talking about the coding strand. They're talking about the coding strand. Upstream, downstream. Okay, upstream 5' end of the coding strand, downstream 3' end of the coding strand. Don't get it confused. Open your NCERT and there is an image given. I think I did not snip that image. Okay, let me go here. Okay, so in this image, is this clear? It will not be that clear, but image will be clear. Okay, don't worry. See this one. See, this is what they're talking about. Yeah, this one. See here? Can you all see here? Should I zoom more? Yeah, visible, no? Visible? Yes, visible. So here students, look here, the promoter is on the upstream end, okay. This is the 3' to 5' end, which is going to act like the template strand. Whatever I told stands the same, okay. Template strand is going to be from 3' to 5', okay, so that the RNA polymerase can form a strand which is 5' to 3'. Now the coding strand. So here they are talking about what? Here they are talking about what? Upstream of the coding strand. That means on the coding strand at the 5' end, your promoter is going to be present. That is, this is going to be the upstream. Okay. Just know this. Don't confuse yourself. Don't think, oh my god, what is this? NCERT is killing me and all. Don't cry. Just look here. 5' end of the coding strand, there is a promoter in the upstream. Okay. Now same way, in the 3' end of the coding strand, you will have the terminator in the downstream. Is this clear? Yes or no? Okay. Upstream means what? Something up on the top position. Now downstream means what? Something in the lower position. That's all. Here they are talking with the help of coding strand. This is what you will not understand when you read NCERT. That is why you need the help. Okay. Clear? All of you, is it clear? Take 2 minutes, try to understand, and I will explain it again if it's not clear. Okay, very simple. Just remember that they're talking about the promoter and terminator in the terms of the coding strand, not in the terms of the template strand. That's all. If you understand this, everything is very clear. Okay, whatever we spoke about the template strand remains the same, whatever we spoke about the coding strand remains the same, just that they're talking about the promoter and the terminator in reference to the coding strand. I want all of your thumbs up in the chat box, quickly. Clear, ma'am? Okay, perfect, perfect. Okay, now we'll go back here. Okay, look here. As it usually defines, okay, all this we are not going to talk about. Now see here, there are additional regularities sequence that may be present further upstream or downstream to the promoter. Some of the properties of this sequence shall be discussed when we talk about gene expression. Now, here comes a very important part which I will only explain, but before that I will just give you a very small idea about this. See, the DNA sequence coding, students, that's why I tell you that this chapter is not very easy to explain with NCERT. See, when I teach, I can explain it very smoothly without, just telling as a story. But when we read NCERT, what happens? There will be so many words that you will not understand, statements that you will not understand, okay? But we will try as well as possible to make it very clear to you. The DNA sequence coding for tRNA or RNA molecule also define a gene. However, by defining a cistron, that is a segment, okay? As a segment of DNA coding for a polypeptide, okay, polypeptide. So students, cistron is a small portion of the strand, okay, small portion of a DNA. So we can also call it as a segment of DNA, okay, that can code for the mRNA and ultimately form the proteins. Because always remember, entire DNA cannot code and form the proteins, right? Only a small portion can, okay. So the structural gene in a transcription unit could be said monocistronic, mostly in eukaryotes, polycistronic, mostly in prokaryotes. Shall I do GM Express for this? All of you? Shall I do GM Express? Okay, because I have confidence in explaining you better, because sometimes if I make you read NCERT, you might panic. Okay, so students, look here. This is a gene. Okay, this is another gene. This is a gene, okay, a small segment, we can call this a cistron as well. Now this cistron, okay, this one is in prokaryotes. In prokaryotes, students, there is something called as coding sequence and non-coding sequence, okay. What is it called? Coding sequence. Coding sequence, that is called as exons. What is that called as? Exons, okay. Non-coding sequence, that means they are not going to code and they are not going to help us in the formation of protein. Okay, so they are non-coding sequence. They are called as introns. Okay, they are called as introns. Clear to all of you? Is this clear? Okay, yes. Now remember that this one is in case of a prokaryote. This is in case of whom? A prokaryote. Clear? Now in prokaryote, the thing is, advantage is they only have exons. They only have exons. No complications, only exons are there, all are going to code, and they are going to code for multiple proteins, as I already mentioned to all of you. So we will call this as polycistronic. We will call this as polycistronic. Anyone has doubt in this? Anyone has doubt in what I just told now? Remember pro, poly, okay, PP. Got it? Polycistronic. That means in prokaryotes, tension is not there. The segment that we took, all of it is going to code for protein. Everyone is happy. Each of it is going to code for multiple proteins. So we will call it polycistronic. Any doubt so far? Any doubt so far? Please let me know in the chat box. What happened? Why are you people telling welcome? Today is my first day. Okay. Why are you people crying? Or are you people randomly putting emojis? Clear? Okay, nothing complicated. Just remember that in prokaryotes, the entire strand, this is a gene. Okay, what I've drawn in this pink is a gene. The entire gene has only axons, that means only coding sequence, and since the entire strand has coding sequence, that gene is very simple. Okay, simple. Now here what happens, student, this is in our case, okay, in eukaryotes, what is going to happen? They have some blockage in between. Okay, they have some blockage in between. Okay. So can you see? I have drawn two different colors. Okay, I have drawn two different colors. I think this color is not that visible. Okay, now two colors are visible. Yes, two colors are visible. Now students, when you see here, this is also a cistron, a small segment which can code for a protein. But here what has happened? They have exons, introns. Exons, introns. That means in eukaryotes, they have a problem. The problem is the presence of split gene. The presence of what gene? Split gene. That means they have regions where they have coding sequence as well as non-coding sequence. Is this clear to all of you? Is this clear to all of you? E means exons, I means introns. Okay, so they have a region where they have exons and introns. That means coding and non-coding sequence are together. Now the problem with this, I will not require the introns at all, because I need to ultimately form my proteins. I have to do translation. So for that purpose to happen, what I am going to do, I am going to remove the introns, and the removal of introns and the adding of your exons together, removal of introns and joining your exons together is called as splicing. What is it called as? Splicing. So in splicing, what is going to happen, students? We are basically going to remove the introns. Okay, we are going to remove the introns from this, from the split gene, so that we can get a gene which is only having exons. Clear? Which is only having exons. So this way, what am I going to get? I am going to get a gene I am going to get a gene that has only exons, and they are going to code for my proteins. Okay, I am going to get a gene which has only exons, that is after splicing. So in this gene, okay, this is after splicing. Okay, after splicing, I am going to get a gene only with exons. Now I am going to do capping and tailing of this. Capping and tailing is where, so that no other extra additional anything happens, and also for translation to happen smoothly, we are going to give capping and tailing. So if I am going to give capping, it is going to be with the help of methyl guanosine triphosphate. Okay, methyl guanosine triphosphate. Okay, that is going to help in capping. Okay, now tailing I am going to do with the help of poly A tail. Is this clear to all of you? Yes or no in the chats? Students, I am not able to raise my voice because of the cracking. That's why I am talking very softly. Okay, yes, I hope it is clear. Fine, it is because of I am going to do the tailing with the help of poly A tail. that is going to do the tailing. So this way, when my gene is going from the nucleus to the cytoplasm for translation, it will be properly coded, right? Clear, all of you? Perfect. Again, we will learn this from NCERT, do not worry, but try to learn this, understand. Now students, because this gene, okay, as I told you, the rule is there where we form from one DNA, one protein, we cannot misuse it. So this is going to be a monocistronic gene. This is going to be what? A monocistronic gene. Clear to all of you? So eukaryotes have monocistronic gene. Cistron is clear, monocistronic is clear, splicing is clear, capping is still clear, yes or no? Yes. All of you tell me, capping, tailing, everything is clear? Any portion that you have a difficulty, please let me know right now. Clear, okay? All of you, can I go back to NCERT? Now you will understand NCERT very, very smoothly. Don't even take tension, okay? Don't even take tension. Look here. Okay, now look here. The structural gene and transcriptional unit could be said as monocistronic. In eukaryotes, the monocistronic structural gene have interrupted the coding sequence. Understood? The genes in the eukaryotes are split. The genes in the eukaryotes are split, so we will call it the split gene. Now the coding sequence or the express sequence defined as exons. Exons are the one who are going to express themselves. Introns are the one who is going to not express themselves. Exons are said to be those sequences that appear in mature or processed RNA. So I am going to teach you what is RNA processing. Now whatever we did is only RNA processing. That means where we remove the introns and where we join all the exons, we do capping and tailing. That is only called as matured RNA or processed RNA. That means you know how your food and all gets processed. That is when you are taking bread and all, you think that, oh, bread is so nice, oh my god, so healthy. They would have written outside 50% wheat, 25% fibers, 50% protein. All that is all processed food. That means everything is coming from companies. Some mixing they will do, manufacturing, everything will happen at one shot. It will come to the market. Whatever they want, they will write it on the cover. Okay, that is up to them. Like, okay, they feel like, okay, I want to write 80% protein because all the gym freak will see that 80% protein and buy the bread. So they can do different marketing strategies. So this is also processing. That means what we want, we process and get it out. So that RNA which I formed here, okay, that RNA which I formed here is going to be called as my processed RNA. Okay, this one is going to be my, this is going to be my processed, okay, processed RNA, okay, or mature RNA. So students, in mature RNA, I am only going to have exons, okay, because I processed it, right? I processed it. Got it? So is this clear? Now, let's go back here. The exons are interrupted by the introns. Introns or intervening sequence do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of gene in terms of DNA segments. So in eukaryotes, everything becomes complicated. Hello Rani. Now, inheritance of a character is also affected by the promoter and regulatory sequence. Hence, the regulatory sequence are loosely defined as regulatory genes, even though these sequences do not code for RNA or protein. We will come back to that. Now, students, see, this is exactly what transcription is. Okay, as I told you already, 3 units are there: promoter, structural gene, and your terminator. Now the process that is going to happen is going to be initiation, okay, elongation and termination. Now during initiation, I already told you that RNA polymerase will take the help of sigma factor. Okay, RNA polymerase will take the help of sigma factor and it is going to start the process. Now where is it going to do? Okay. Okay, where is it going to do? Go, like, it is going to go to the middle. That is where the structural gene is present. There it is going to form. This yellow color which is coming out is the mRNA. Okay, yellow color which is coming out is your mRNA that is formed. Clear? Now students, after some time the sigma factor is going to go. Now the RNA polymerase does not know where to stop. RNA polymerase does not know where to stop. So to make sure that the RNA polymerase stops at the right position, we have a rho factor which is going to combine in the terminator region and terminate the entire reaction. That's all is your transcription. Okay, elongation, initiation, sigma factor plus RNA polymerase will start the reaction. It will go exactly at the structural gene region, form the mRNA, that is elongation. Now after that, the RNA polymerase does not know where to stop, so it will go and bind with the rho factor. Rho factor will terminate the entire thing. Okay, terminate the entire thing. Now remember that students, RNA polymerase can be of three types. Okay, I always told you this example, RMT. Okay, RMT, which one of my teachers taught me. So RMT, no, one of my seniors taught me. So RMT students, write 1, 2 and 3. Okay, so rRNA is going to be formed. Okay, rRNA is going to be synthesized by RNA polymerase 1. Okay, this is all your RNA polymerase. Three types of RNA polymerase is there. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. RNA polymerase 1. Okay, RNA polymerase 1 is going to synthesize rRNA. Again, RNA polymerase 2, mRNA, RNA polymerase 3, tRNA. RMT, like MTR, remember RMT. Clear to all of you, yes or no? Clear? Okay, fine. Now, this is your transcription. Now here students, again few things I explained about the same thing. The RNA polymerase is only capable of catalyzing the process of elongation. That means what? It associates with initiation factor, which is your sigma factor, and termination factor. That means RNA polymerase technically can do only one work, that is elongation. That is with the help of structural gene it can form the new mRNA. Rest everything is happening because of sigma factor and your rho factor. Understood? To initiate and terminate the transcription respectively. Association with these factors alter the specificity of RNA polymerase to either initiate or terminate. In bacteria, since the mRNA does not require any processing, that means in prokaryotes we do not need to do RNA processing, so it is going to become active, and also transcription and translation takes place at the same compartments. Okay, same compartment. So students, here they are talking about who? They are talking about prokaryotes. They are telling that in prokaryotes we do not need RNA processing. Yes or no? Do we have to form mature RNA? We do not have to form mature RNA because they have introns and exons, right? They do not have introns. So in prokaryotes there is no need for RNA processing. Okay, no RNA processing required. Since we do not need RNA processing, we will not have the tension at all. Okay, we will not have the tension at all. So look here, compartment, there is no separation of cytosol and nucleus in the bacteria. So students, always remember the entire process up to transcription is going to happen in the nucleus, translation is going to happen in the cytoplasm. So in ours, after we remove the split gene and everything, we are going to form a new mRNA, proper capping and tailing will be done, and then we will send them to cytoplasm. But here such a difference is not there. The nucleus and the cytosol, such a difference is not there, so everything will happen simultaneously. Clear? So here there is no separation of the cytosol and the nucleus in the bacteria. Many times the translation can begin much before the mRNA is fully even transcribed. Consequently, the transcription and translation can be coupled in bacteria. Is this clear? So the biggest help of reading NCERT is such kind of statements will never be missed. Super clear, students? Anyone has any doubts? Okay, now students, this is the processing which I spoke about. See, this is the mRNA that formed. Can you see? We have introns, exons, introns, exons. Now what are we going to do? We are going to remove the introns with the help of splicing, RNA splicing, where we will remove the introns as well as we will join back the exons. Okay, and then we are going to form the newly formed mRNA, where we are going to do capping at the 5' end and we are going to do tailing at the 3' end with the help of poly A tail, and capping is going to happen with methyl guanosine triphosphate. You can see here MGPPP. PPP, remember, nucleoside triphosphate, PPP. Okay, triphosphate, that will give you a messenger RNA. Any doubts so far? Any doubts so far? Okay, clear? Super clear? Yes? Okay. Now students, look here, whereas the RNA polymerase 3 is responsible for the transcription of tRNA, this question has come many times in NCERT, for NEET. Okay, many times this question has come as match the following, as a statement based question. So look here, RNA polymerase. Remember RMT. Do not forget this. Remember RMT. Any questions from RMT can come. Okay, RMT 1, 2 and 3. RMT 1, 2 and 3. Any question can come. So, the RNA polymerase 2 transcribes the precursors of mRNA, forming the heterogeneous nuclear RNA. Okay, heterogeneous nuclear RNA. Again, from the word hetero, you understand two different. That means exons and introns will be there. Okay. What is Kohinoor plus Gopika? Okay, the second complexity is that the primary transcripts contain both exons and introns and are non-functional. Hence, we do splicing and splicing. So, same thing students, here it is already given. See, exons are joined in a defined order. So, remember that splicing is not just removing of introns, it is also joining back of exons. Okay, done. All of you clear? Fully clear? No doubts? Any doubts? Do you want a break now? Brain is braining or brain is not braining? Huh? We'll take a break, chuttu break we'll take and come back, okay? Students, pray that my throat works till the end of this class, okay? This is the problem, teacher's problem, that throat will give work every then and now, okay? We'll take a small break, 10 minutes break, we'll come back, I'll give you a lunch break after some time, okay, because now it's only 12:59, I'll give you a lunch break after some time, like when it's 2 o'clock, I'll give you a lunch break. Right now, you take a small break and come back, so even my throat will have some rest. Okay, 1:10, okay, scooty break. Yes, okay. Yeah, Rani, that's because you joined now. These people are sitting from 11, no, their brain is not braining. Is it lagging? guys. Or weather forecast in the chatter. Is this one better? Now the reflection is better, right? Too much reflection was coming, no? So I just asked them to fix that. Okay, we'll continue. Okay, now students, we have learned transcription. Now the next topic. Okay, ready all of you? Perfect. Now the next topic that we have is genetic code. Okay, now genetic code, there's no much explanation we have. We have a box and we have some salient features. But other than that, there's a general, some introduction part that is there. So students, genetic code was actually a difficult thing to land in. Like now we are learning it so easily, but it was not this easy. Okay, but it was not this easy. Clear to all of you? All of you come back to the class? Yes, okay. Now see students, genetic code wasn't that easy to find, or it was not very easy to land in this code that the code is going to be triplet. It was not so easy. So what they did was they started where they were like, okay, see, we have this many things in our hand, that is A, T, G, C. This is what we have in our hand. Okay, these are the bases that we have. Now with the help of these bases, we have to make a code. Okay, we have to know that on the mRNA there is, let's say this is the mRNA that we formed. Okay, this is the mRNA that we formed. This is the mRNA we formed. In this mRNA we have different codes, A, G, A, T, T. Okay, now this is the mRNA, no? So U, okay, C, C, A, G, U, something like this we have code. Now how do I know that AGA is going to code for an amino acid? For that I have to I'm so sorry yaar, I forgot to wear the mic. Okay, now it's fine. Okay, now it's fine. I don't know today why so many technical issues are coming. Okay, yes, now it's fine. I'm so sorry, I forgot. Okay, now what happens students? So what I was telling, genetic code wasn't easy to land in. Okay, they had to go through a lot of finding to land to this place. So what happened, the only thing that they had was ATGC, that is 4 bases that they had. With the help of 4 bases, they were like, we have to find out which protein are they going to code for. So they were like, how do I know AGA is going to code for which protein, so or which amino acids? Okay, amino acids. So for that they kept finding new things. Okay, they kept finding new things. So genetic code started with the help of, they were like, okay, let's do one thing, let's take them individually, that is 4 raised to 1. Okay, that will give 4 nucleotides, right? 4. So it can code for 4 amino acids. So they have told that we have 20 amino acids to code. Okay, we have 20 amino acids to code. So you people please find code for it. Now they started with 1 each. That means A will code for 1, T will code for 1, G will code for 1, C will code for 1. It is not working out. How many amino acids can we code for? Only 4 amino acids. Yes also. Yes. Oh, wow. Your people's memory? You tell me about your memory. Without revision, if I make you do one question, you will forget everything. Right? Everything you will forget, and you people are talking about other people's memory. No? Very funny, very funny. You people can't even remember, is it the 5th thoracic vertebrae the trachea is dividing, or in the 7th thoracic vertebrae? And you people are telling about my memory, that I forgot the mic. Okay? Very smart. If I start checking your memory, you will cry. So let's not get in there. Okay, let's not get into a region where we are not, okay, having a nice time. Fine. Agreed? All of you? Oh, they see, easiest way is they accept that ma'am we are 60 years old. Very smart my students are. Very smart. Very smart. Okay, there's no one as smart as my NEET English students. Okay, come back to class now. Look here, they tried 4 raised to 1, did not work. We were able to only code for 4 amino acids. Then they told 4 raised to 2. Then they were like, okay, let's do one thing. 8 amino acids we'll be able to, or let's say 4 into 4, no, so 16. Okay, 16 amino acids we'll be able to code for. Still we have how many? 4 amino acids are still missing. Okay, 4 amino acids are still missing. So we cannot code again. So then they were like, okay, fine, let's do one thing. Let's make combinations of 3. Okay, combinations of 3. Even though it is going to help us code for 64 amino acids, even though we have only 20 amino acids, more is better. Less is problem. More is better. Okay, more is better. Yeah. So now they went for this permutation and combination where they got 4 to the power 3 that helped them code for 64 amino acid. Now what is 4 to the power 3? They were like, if there are 4 bases, we are going to make a combination which is going to be a triplet combination. Okay, it is always going to be 3 people together, be it AUG, be it UUU, okay, be it UGA, all of it is going to be in triplets. They told that out of 64, okay, out of 64, 61 is going to code for amino acids and 3 of them are not going to code for anything. We can call them the stop codons. We can call them what, students? Stop codon. So the introduction is clear, and this was found by a physicist, okay, called as George Gamow. He was the one that told that let's go for this permutation and combination. Clear introduction? Clear introduction? Yes. Okay, so now we will go to the code. Luckily, you do not have to learn this entire box, but just know how are we getting these 3 codes. So students, normally in your Punnett square, you study 2 combinations, that is A, that is two positions will be there. Now here you can see 3 positions are there. So U, C, A, G will be written in 3 different ways, okay, 3 positions. That is going to be U, 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 okay, this is the 3rd position, okay, U, U, C, U, U, A, U, U, G, got it? U, 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 C, U, U, A, U, U, G, okay, understood? Now which are the stop codons, ma'am? It is going to be U, A, stop, U, A, G, stop, okay, and one more is going to be UGA, which is going to be stop. These 3 are going to be that person who is not going to code for anything, stop codons. Clear to all of you? Is this clear? Very simple it is. Now salient features you have to remember, okay, salient features you have to remember. See, the salient features also are not that complicated, like you cannot study, it's very simple. First thing is the codons are always going to be in triplet, that means 3 of them together. So the codon is a triplet out of which 61 codons will code for amino acids and 3 codons do not code for anything. That is why they will be called as stop codons, okay, they function as stop codons. Some amino acids are coded by more than one codon, hence the code is degenerate. Students, here only you can see here, see your again let's take, okay, let's take lysine, because you have heard the word lysine and arginine. Okay, let's take lysine. Is it buffering? Don't tell chuma chuma. Okay, tell the truth, is it buffering? I can't see any buffering. Stop codons, okay? Hey, no, chumma you people are telling. Okay, listen here. Let's take lysine and arginine, students. Now what's going to happen? Look at lysine, AAA and AAG codes for lysine. Same thing for arginine. So there are most of the amino acids there where they have more than 2 codes that are coding for them, okay, coding for them. So here that's why we tell that some amino acids are coded by more than one codon, hence the code is degenerate. Next one, the codon in red in mRNA is in a contiguous fashion, that means in a continuous fashion, okay. That is, if I have to read a codon that I am writing AAA, AGA, UUU, UUA, okay, this is the codon I have written. I will not read AAA, comma, AGA, comma, UUU, comma, I will not read like that, okay. I will read it in a continuous fashion, starting from A, ending up to A, I will read it in a continuous fashion. Understood? Now, the code is nearly universal. That means the code is nearly universal. That means what? Say it even in bacteria, even in an elephant. If here they have given UUU will code for phenylalanine, it is going to be the same even if it is a bacteria, even if it is an elephant. That means from the smallest organism to the largest organism, everyone is going to be universal. Okay? Yes. Clear? You do not have to by heart the table. You do not have to by heart the table. Luckily, no questions will be asked for the table, only very common ones like your methionine, which is already given here, that you have to remember. You do not have to by heart the table. Don't go and by heart the table. Okay, don't go and by heart the table and waste your time. Now look here. So for example, some bacteria to human UUU will code for phenylalanine. Some exceptions to this rule have been found in mitochondria codon in some protozoans. Again, not your tension, don't take it up. Okay, so UUU will code from bacteria to human, the same thing. Phenylalanine only is going to code. UAG has dual function. That means it is a start codon, at the same time it is also going to code for methionine, depending on where it is going to be present. If it is present in the initial parts, it's going to do initiation. That means it is going to help in the start. But if it is present in the middle, it is going to code for methionine. Clear? So it is going to act as an initiator codon. Clear? Initiator codon. Now UAA, UAG and UGA are terminator codons. Okay, see the combination, students. UAA, UAG, UGA. Okay, UA, Yug, Yuga. Okay, it's like Nayanthara's kids name, no? Something like this, only their kids names, no? Well, if you remember, some Yuga, something like that only. So UA, Yug, Yuag and Yuga. Okay, done. Salient features are easy. Yes or no, all of you in the chat? Very easy. Okay, simple words are there. First thing is they are triplet. 61 will code for amino acids, 3 are going to act as stop codons. Second one, they are going to be degenerate. That means in one amino acid can be coded by multiple codons. Third one is going to be codons are universal, be it from bacteria till humans. UUU will code for phenylalanine. Fourth one is going to be, you are going to read it in a continuous fashion and contiguous fashion. That means without any punctuations, you are going to read the entire codons. Fifth point is going to be your stop codons. UAA, UGA and UAG are going to act as a stop codon. Clear? And AUG has dual function, where AUG will act as an initiator codon as well as going to code for methionine. Okay, Uir and Ulag, right? Yeah, very nice names, no? Okay. Now the relationship between genes and DNA are best understood by mutation. So students, as I told you, we have already discussed, if one thing changes that side, this side, it can cause mutation. Yes or no? Clear to all of you? Okay, so here they are talking about sickle cell anemia, students. You can see that glutamate to valine. Okay, glutamate to valine, that is your GAG. GAG, this A got replaced by GUG, right? So because of it, glutamate got coding to valine, that led to the formation or led to the sickle cell anemia. Again, a point mutation. That means at one change with one base can change the entire combination. That is why they tell that DNA polymerase have to be highly accurate. Clear? This is just one example here. Okay, one example, glutamate. This was supposed to code. Okay, this was supposed to code for glutamate, students, but it U happened, so it will code for valine. Done. Now insertion and deletion. Okay, insertion and deletion. Any doubts so far? Everything is super duper clear? Any doubts? My speed is fine. Okay, now look here. Ram has red cap. I've taught you this many times and it is very easy. Now students, someone has added a B here. Okay, someone has added a B here. So here what is happening? It is an insertion. It is an insertion. Yes or no? One B has been added here. So here they are telling us that if we do insertion, if we do deletion in between a codon, what is going to happen? It will lead to mutation. It will lead to frame shift mutation. Now imagine I am reading Ram has red cap. Suddenly one B came there, I am not able to read anymore. That means what? My reading shift changed. I can tell, no, my reading, I am not able to read anymore. Same way students, in the genes, always remember frame shift mutation happens. Okay, now the reading frame, how it got shifted for me, same way for them also coding will change. Okay, now they will code for the first 3 triplets. For them they will not read if it is correct or not. So they are going to start coding for whatever is left. So this will lead to mutation. So if we are inserting something, it has to be a triplet. If we are deleting something also, it has to be a triplet. Clear to all of you? Yes or no? So look here, Ram has red cap. Here we inserted a B here. Now because of the insertion of B, what is going to be read? It is going to be read Ram has BREDCA and P will be left alone. Now let's say we inserted one more letter to it. Okay, we inserted one more letter to it, that is we inserted I to it. If we insert I also, you can see that the reading frame is the same. Ram has BIREDCAP. Okay, now again it is going to read whatever is left over, it is going to read like this. Again we have not reached to a proper reading frame. Now let's say we decide to add one more letter to it. Now you tell me, is the reading frame correct or wrong? All of you in the chats, let me know if the reading frame. I left my water bottle. Clear? Yes or no? Tell me, is it correct now, students? Has the frame shift happened? Has the mutation changed? Yes, has it changed? Yes, ma'am. Very good, very good. Yes, ma'am, it has changed. Correct? That is, Ram has big red cap. Ram has big red cap. So now it is making more sense. That is, we are able to read exactly Ram has big red cap. Now my reading frame has come back to its original state. No? Now see students, the same exercise can be repeated by deleting. Same thing I can do. If I delete one letter, it is going to not make any more sense. But if I delete 3 letters together, it was going to make Ram has cap. So when we are adding, okay, remember this, when we are adding, okay, or deleting, we have to make sure that we are doing the triplet rule. Okay, that is, triplet will be added, triplet will be deleted. Clear to all of you? All of you? Yes, so far good. Any doubts? This will lead to frame shift mutation. Okay, so here only I'll write that. This will lead to, okay, frame shift mutation and insertional mutation. Okay, again, insertional mutation. Done, done. Insertional mutation and frame shift mutation. Clear to all of you? Can we go to tRNA? Yes. So students, look here. Now, as we already understood, code is there, ma'am. Everything is there. Now we understood so far clear that on the mRNA there are going to be your bases. Now when we read it as triplet, it is going to code for an amino acid. But do you think our own mRNA will be able to read code for amino acid, make the polypeptide chain? No. Right? We cannot do all of this. Our mRNA cannot read the code, then tell that, okay, AUG, you are going to code for methionine, then add methionine, then form the peptide bond, then make an entire polypeptide chain is too much of a work. So, even though RNA is not a genetic material for 99% of organisms, we know that RNA has catalytic nature, RNA can act as an adapter, RNA can also act as a messenger. So we are going to take the help of adapter RNA, which is a tRNA, who is going to read the code on the mRNA and attach the amino acid according to it and help us form the polypeptide chain. Clear to all of you? So the adapter molecule that I am going to take is a tRNA adapter molecule. So, from the very beginning of the proposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also to link it with the amino acids. Clear to all of you students? Since our mRNA cannot do all of this task alone, we are going to take the help of another person. Okay, we are going to take a help of another person because amino acids have no structural specialities to read the code uniquely, or else the other option is that the amino acids itself read. Amino acid will be like, okay, AUG, methionine come in code, I have to come in code. Next one will be, okay, valine, GUG, valine, I will come in code. They do not have any structural speciality where they will only come, read and attach themselves. So these two options were nil. So we took a help of the tRNA. He postulated the presence of an adapter molecule that would one hand read the code on the mRNA, okay, code and on the other hand will bind the specific amino acid. Clear to all of you? Easy peasy lemon squeezy. So in one side it is going to make sure to read the code, okay, the genetic code. Whenever I am telling code, remember the genetic code, and on the other hand it will attach the amino acids. Okay, it will attach the amino acids. Clear? Perfectly okay? Perfectly okay? Okay, done. Now, the tRNA, then called the sRNA, that is the soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. Okay, now students, this is exactly what is going to happen. Then again, I will go back to NCERT. So look at this. This is your mRNA. Okay, this is your mRNA. On the mRNA, there is code. Can you see? AGU is the code on the mRNA. Now, tRNA has different loops. Okay, different loops. First loop which is there, which is going to be very close to the mRNA, is called as the anticodon loop. What loop is it? Anticodon loop. This anticodon loop on the tRNA is going to make sure that it forms anticodon. That means if it is, look here, if it is AGU, it will form UCA. Okay, UCA. Now, on the other end of your tRNA, they have an amino acid acceptor site. What do they have, students? Amino acid acceptor site. Understood? Acceptor site. So, in one side, one is going to do the anticodon job, one is going to start adding the amino acid, which our amino acids cannot do, our mRNA cannot do. That is why we took the help of tRNA. Clear? Easy peasy? So much clear, no? Now, always remember that, students, there are sites on the tRNA. There are sites on the tRNA which are called as your EPA site. EPA site, where E is going to be your exit site, where you will exit. P is going to be where your protein synthesis is going to happen, or your peptides are going to be added. Clear to all of you? Where your peptide formation, its addition is going to be happening. Okay, so this is going to be a peptidyl formation, okay, area, and A is going to be your acceptor site. Okay, acceptor site. Now remember that if an amino acid is coming, okay, if an amino acid is coming, always remember that it is going to be first entering into the acceptor site. Okay, let's say one tRNA is there, the tRNA is bringing, let's say letter A. Okay, then after that it will go to the P site where it will form the peptide, then it will exit to the E site. Clear to all of you? Yes or no? Clear? There's a diagram, I'll show you the diagram. Okay, I'll show you the diagram. Now, let's see before that. Okay, now you understood about tRNA, but I will show you the diagram from your NCERT. Okay, diagram from NCERT. Okay. Okay, here genetic code. Yes. Students, look here. This is the diagram, one diagram which is given. Then we'll go to translation. See, look here. Okay, now that I'm speaking about tRNA, I'm going to explain this also to all of you. Okay. Now, students, look here. This is your tRNA. I think if I zoom so much, it will be too much. Okay. Now, clear to all of you? Diagram is clear? Can you see the diagram? Yeah, it's visible, no? Now, students, look here. As I told you, this is the mRNA. On the mRNA, all of you can see the code? Yes, ma'am, we can see the code. Right? Can I enlarge? Oh, yeah, I can. Okay, yes. Students, this is the code. Now, can you all see there are sites? 3 sites are there. See, look here. This is one site. Okay, this is, why is this not writing now? Okay, 1 second, let me try again. Yeah, this is one side, this is one side, this is one side. Now, all of you listen to me carefully. Can you see one person waiting to enter? So, this is where your acceptor is going to come in place. Okay, acceptor site is going to come in place. That means one person is waiting to come and join here, and this tRNA has the anticodon of this one, AAU. AAU. What is it going to code for? UUA. So, can you see anticodon is already ready. All of you open your NCERT and keep if this diagram is not clear. Okay, if this diagram is not clear, open your NCERT and keep, and let's see. See, AAA is waiting down in the mRNA. They have already formed the anticodon for this. Okay, anticodon for this is already formed, and it has already coded for the amino acid as well. It has coded for the amino acid as well. Now, it will come to the acceptor site, it will wait, and then what it is going to do? See, can you see this arrow mark towards this? Now, this whole polypeptide chain will start joining here. Will start joining here. Now, when this fellow comes, this will be shifted to this person. So, remember students, from here, this is going to be the exit site. Can you see one person has one person exited from this site. Okay, this will become a meme, I feel. Okay. So students, clear to all of you? Is this clear? Okay. So, what is going to happen? This is going to be the acceptor side. This is where the polypeptide chain will be formed. This is going to be the exit side. So, you can see after this was added, this person is exiting with the tRNA. The next person is waiting to come in. As soon as this person attaches this chain here, this person will also exit. This person will move this side. This person will come and join. So, you understand, like one seat get over, another seat will come. One seat person get over, another seat will come. So, it is going to keep happening like this. Okay. One seat is over, next person will shift like this. Then that person will exit, another person will be waiting to come in, exactly like this. Okay, how your queue system happens, no? When we go to take Aadhaar card, PAN card and all, how it happens, one person, 2 people will be waiting to take the photo. Okay, one person would have already got up, then one person will be already waiting with all the documents in their hand. Same thing is going to happen here. They'll be ready with the anticodon and the amino acid acceptor side, everything will be ready. They'll be waiting with all the documents. The moment this person gets up, this person will move this side and this person will come and sit. Same thing, but here sites are there, E side, P side, A side. A side is the acceptor side, P side is where the polypeptide chain will be formed. Okay, and the E side is the exit side. Is it clear? I want thumbs up in the chat box. All of you tell me thumbs up in the chat box, or tell easy peasy lemon squeezy. Okay, easy peasy lemon squeezy will take a lot of time to type. So, is this clear? Okay. Yes. Don't make a meme out of it. That's it. Okay. Yesterday only I saw a meme when people forget English suddenly. Okay. Yes. Thank you, Hari. Okay. This is perfectly clear, because we'll come back to translation now. Okay. We are going to come back to translation now. So you should be clear about this. Otherwise, you will not understand this. All of you. I don't want just 1 or 2 people telling it. I want everyone to tell it. Okay. Easy peasy. Okay, fine. Now students, let's come to translation. Okay, translation refers to the process of polymerization of amino acids to form polypeptide chain. So we already know that according to our central dogma, right? According to our central dogma, we know that when RNA becomes protein, that process is going to be called as translation, right? That process is going to be called as translation. Now, in translation, we are going to take the help of some people, as we discussed right now. The people that we are going to take help is going to be your tRNA, and students, something that I did not tell you is ribosomes. Okay, ribosomes. Remember that your entire translational unit happens with the help of ribosome and your tRNA, which I will show you in NCERT. That's why I did not discuss right now. But remember that when your smaller subunit of ribosome comes and joins, then only the entire thing is going to continue. That means what? Let's say if one of you, let's say Sukanya hasn't liked the video. Yet still now Sukanya hasn't liked the video. That means what? So the class has to start, or let's say we have kept a target of 200 likes and I have told you that only after 200 likes I will start the class. So Sukanya is that last person to like. The moment Sukanya likes the video, it will become 200 and then I will start the class. Same way, until the smaller subunit comes and joins and forms this entire translational unit with tRNA, larger subunit of the ribosome, until the smaller subunit come and bind, the entire translation will not start. Clear to all of you? Clear to all of you? So here again, that will going to come to your notice in NCERT. Okay, now we'll start translation. Ready? Energy is there? Okay, energy is there? All of you? Tell me. Some of you haven't still liked the video. Who is that? Misbah liked the video. Okay, so clear? Can I continue? Now, see students, translation refers to the process of polymerization. That means from RNA, mRNA that we formed already in transcription, we are going to read the code and attach the amino acid. Amino acid is going to form the polypeptide chain and that is going to help us in all of it. Okay? Yes. Now students, look here. The order and the sequence of amino acids are defined by the sequence in bases. Okay, in the mRNA. This we already know from genetic code. The amino acids are joined by a bond which is called as a peptide bond. Yes or no? If many amino acids, let's say this is an amino acid, it has to combine with this amino acid, this bond which is going to be between them is going to be called as the polypeptide bond. Polypeptide bond. Clear? Now, therefore, in the first phase itself, amino acids are activated in the presence of ATP. So, first thing is, if you want to form the peptide bond, you require ATP. If you want ATP, you have to charge the entire process first of all. Right? So, if you want to form peptide bond, first you need to charge your tRNA. tRNA has to get charged. With the tRNA getting charged, the whole process is going to continue. So, listen to me carefully here. See, formation of a peptide bond requires energy. Okay? Therefore, in the first phase itself, amino acids are activated. Okay, in the first phase itself, amino acids are activated in the presence of ATP and linked to each other. So, students, only if the amino acids are getting activated, it is going to form the peptide bond. Okay, it is going to form, very good, form the peptide bond. So, tRNA, that is a process commonly called as charging of tRNA or amino acylation of tRNA. That means the first ever process is I need to form the peptide bond because many amino acids have come together. Yes or no? Amino acid, many amino acids have come together. Now, if I want to make sure that these amino acids are going to come together, I need to make sure that there is a bond, and that bond is going to be none other than your peptide bond. This peptide bond formation requires energy. So, my first process will be to charge the tRNA or do amino acylation of tRNA. Clear? Yes? Yes, clear? Any doubts in this? Okay, done. Now, to be more specific, if two such charged tRNAs are brought close enough, the formation of peptide bond between them is going to happen. Clear? Okay, peptide bond between them is going to happen. So, we bring two tRNAs. Let's say this is our one tRNA. Okay. Okay, this is one tRNA. Students, tRNA looks like clover leaf model. Okay, this is one tRNA. We bring one more tRNA near to that, and let's say this is also charged. Okay, this is also charged. Okay, this is also charged. Easy to form a peptide bond between them. That is what they were talking about. Okay, chalo. Now, would be favored energetically. The presence of catalyst would enhance the rate of peptide bond formation. Now, the cellular, again, there are I have taught in previously about AMP and all of that, but right now I do not see a lot of, okay, brain is not braining her. Oh my god, already, okay, let's finish up to translation, okay, let's finish up to translation for today. So all of you listen till that, okay, all of you listen till that, because it's very important. Translation, Lac-Operon also we have to finish, okay, Lac-Operon also we have to finish. Okay, now let's come back. So students, look here, both the tRNA first processes charging. Why do we need to charge? So that the peptide bond can be formed. Again, there are enzymes which are used, again AMP, monophosphate, many formations are there, which I am not teaching you right now. So understand that charging of tRNA and amino acylation of tRNA is the first step, okay. Now the cellular factory responsible for the synthesizing protein is ribosome. This you have studied in cell chapter. The cellular factory who will synthesize the proteins is the ribosome. The ribosome consists of structural RNAs and about 80 different proteins. Now, it is in its inactive state. It exists as two subunits. Always remember, your tRNA, your ribosome will have a larger subunit, okay, and a smaller subunit. Okay, on these subunits only your EPA sites will be also there. Okay, like this sites will be there, EPA sites. So one will come. So students, always remember, entirely ribosome plus your tRNA together forms the entire translational unit, not just one by one. Okay, so students, once this smaller subunit, so this is the larger, okay, this is the larger, this is the smaller. Now this larger subunit, it's okay, fine, okay, it will already come and bind with the tRNA. The moment the smaller subunit come, the translation is going to start, okay, translation is going to start. Clear to all of you? Clear to all of you? Yes or no? Okay, clear. Now the larger subunit and the smaller subunit. So when the small subunit encounters an mRNA, the process of translation of mRNA to protein begins. This question was asked in one of the years, that's why I am stressing so much. It's an important question. Okay, it's an important question. Remember this carefully, that once the smaller subunit of the ribosome encounters the mRNA, the translation process is going to start. Clear or not? If this question comes, you can thank me next year. Okay. Therefore, there are two sites in the larger subunit for subsequent amino acids to bind to, thus be close enough to each other. So students, again, your smaller and larger subunit, in eukaryotes it can be 80S and in prokaryotes it can be 70S. Right? I think, is it mentioned in NCERT? It's not mentioned, no? Okay. But yeah, remember that S stands for Swedberg's unit, not the size. Okay, don't be like, ma'am, why is it 50 and 30? Okay, so remember 70S will be divided into 50S and 30S. So 50 plus 30 is 80, but it is 70S. So S stands for Swedberg's unit, not the size. Okay, not the size. Don't be like, ma'am, 50 plus 30, what maths are you doing? Okay, so it is not that unit. Now continue. So students, here we are going to start with translation. Remember, the ribosome also catalyzes, acts as a catalyst in 23S RNA in bacteria and enzyme ribosome for the formation of peptide bond. Do not worry about it. Now from here, main portion. The translation unit in mRNA is the sequence of RNA that is flanked by a start codon. Start codon is going to be who? AUG. And the stop codon. Okay, it can be any of the stop codons. It can be UAA, UAG or any of it. Okay. Yes, sedimentation coefficient or your Swedberg's unit. Okay. Now they code for a polypeptide, and mRNA also have some additional. Okay, mRNA also have some additional sequence that are not translated, and they are referred to as untranslated regions. So students, at the end of the mRNA, there is going to be some of the regions which are not going to get translated. Why that regions are there? To increase the efficiency of translation. With this untranslated regions, they are also going to have release factor, which is going to help in end of which is going to help the translation to stop. Okay. Yes. Clear to all of you? Not like every time maths logic is not applicable, but sometimes logic and logic. Okay, sometimes logic is there, students. Okay, you cannot tell always logic is not there. I will not agree to that. Okay, I taught you, in that oxygen dissociation curve graph and all. Guys, next stages series is already live, so go and check it out. Okay, go and check it out and mark your attendance there as well. Okay. Okay, clear? This is clear so far? Pakka? Now don't tell that later it was not clear. Okay, later, now only it should tell me if it's clear or not. So far good, no? Okay, fine. Students, last time I had drew diagrams of your tRNA translation. So now if you want, you can check that video out. Okay, and also I will, the notes are already on the Telegram channel. Okay, it's already live on the Telegram channel. The notes are already there. Okay, let's continue. So the UTRs are present. This is important. They will ask you where the UTRs are present. The UTRs are present at both 5' end before the start codon and 3' end after the stop codon. Okay, I will write all this. Don't worry. I will make a GM Express now and I'll make you write all this. For initiation, the ribosome binds to the mRNA at the start codon that is recognized only by the initiator tRNA. So there is an initiator tRNA which is going to start the process, and that initiator tRNA will be able to read the code AUG and understand that I have to start the process. Okay, now the ribosome proceeds to the elongation phase of protein synthesis. During this stage, complexes composed of an amino acid linked to the tRNA will bind to the appropriate codon in the mRNA by forming the complementary bases with the tRNA, which I already showed you in the image. Okay, the ribosome moves from codon to codon along with the mRNA. Amino acids are added one by one, translated into polypeptide sequences, and this is important. At the end, the release factor binds to the stop codon, terminates translation and releases the complete polypeptide chain. Okay, can we do GM Express of translation? All of you? Yes or no? GM Express? Quickly. What are you people talking? At this point, I don't understand your languages also. Yes or no? We can't understand your language, right? Okay, let's do GM Express, quick GM Express, okay? Very quickly, we are going to just understand what we just learned about translation. Okay, so here, I'll just draw one set, students. Because if I draw many sets, then I will not have time to finish explaining only. Okay, now look here, the one what I have drawn is your mRNA. What I have drawn is your mRNA. Now on the mRNA, you know that ribosomes have to come and attach. Right, ribosomes have to come and attach, and on the ribosome again this EPA site we will draw. Okay, EPA site, EPA site we are going to draw. Now remember that this is from the 5' to the 3' end. Now this is a combination. Now when with this your tRNA also will be there. When the smaller subunit come and bind, okay, when the smaller subunit come and bind, automatically translation is going to happen. Okay, now students remember that initiator tRNA is going to be there. Okay, initiator tRNA is going to be there. I am going to draw tRNA like this. This initiator tRNA will be able to read the codon AUG, and one end of the tRNA is going to have an anticodon loop. The other end is going to have an amino acid acceptor site. This is clear to all of you, yes or no? So at the one end of the tRNA, this is one end of the tRNA, this is going to have the anticodon. Okay, this is going to have the anticodon. That means, let's say on the mRNA we had, okay, on the mRNA we had A, G, U. Okay, this is going to code for what? A will code for U, G will code for C, and U will code for A. It will form the anticodon. Now say imagine UAC is going to code for some amino acid. Okay, can you all check in the box and tell me UCA will code for what? Okay, let's say some amino acid is going to code, I am just writing A. Like this, it is going to continue and it is going to form the polypeptide chain. Now students, the first ever step for the formation or for translation to happen is charging or amino acylation. Amino acylation. Amino acylation of tRNA or charging of tRNA has to happen so that between the amino acids the polypeptide bond, the peptide bond can be formed. Clear to all of you? The peptide bond can be formed. So, for the peptide bond to be formed, we need ATP. For the peptide bond to be formed, I need ATP. For that ATP, I need to do amino acylation of tRNA or I have to do charging of tRNA, right? Yes, Madhumati, very disappointing. Okay, charging of tRNA. Now remember, to charge the tRNA, if I keep it closer, it is easier for the bond formation. tRNA, this is done. Now, after that, what did they tell students? They told that there is going to be the presence of an initiator tRNA, okay, initiator tRNA, which I mentioned there, okay, initiator tRNA that can read the code AUG, okay, that can read the code AUG and start the whole process. Okay, start the process. And for the starting of the process, we also need to understand that there is ribosomes that is going to take place. Okay, ribosomes are also going to be a part of it, and this ribosomes will have two units, which is the larger subunit. Again, this was a previous year question, so you cannot forget this, larger subunit and smaller subunit. Smaller subunit. This larger subunit is going to be already there here. We can see the side, but the moment the smaller subunit comes and binds, the translation is going to happen. Any doubt so far? Any doubt so far? Is this clear to all of you? Now, one more thing that they told is, let's say this is your mRNA at the 5' and the 3' end. They told in the 5' end, before the start codon, this is your, okay, this is your AUG, okay. They are telling that before the start codon, there is going to be UTRs that are present, which is untranslated, okay, regions, untranslated regions, and they are telling that after the stop codon, let's say here the stop codon is there, which is UAA. After the stop codon in the 3' end, there is going to be another UTR, okay, another UTR. Why is this UTR present? To increase the efficiency, to increase the efficiency of translation, okay, increase the efficiency of translation. Clear? This is clear so far, good? Now, they are telling that after the UTRs also, there is going to be something called as the release factor, okay, something called as the release factor that is going to terminate the translation, that is going to terminate the translation. Clear to all of you? Is this super duper clear to all of you? Terminate the translation, okay. Thank you so much, Carbon. Okay, this is going to terminate the translation. Now, release factor will terminate the translation. Other than that, what is there? Amino acylation is there, charging of tRNA is there, initiator tRNA is there, AUG is there, which is going to act as a start codon. At the same time, it's going to code for methionine. But who is going to understand that? Your initiator tRNA is going to understand. And until the smaller subunit comes and binds, the entire thing is not going to start. So, smaller subunit has a very big importance. Okay, done. Now, EPA side, as I told you, just like your Aadhaar card thing, one person will be waiting here and that person would have already kept all the documents ready, not there, come there and then search for the documents. That means, what I am trying to say is, your tRNA that is there would have already made the anticodons, already attached the amino acids, and then it would be waiting to come into the acceptor side. Here the polypeptide chains will be formed. Okay, here the polypeptide chains will be formed, and then this person will be ready to exit. One person will be ready to exit after attaching. Okay, after attaching this. Clear? Perfect. The train you can't see because of the train, no? It's okay. Super clear to all of you? Throat is gone.
Someone put the AC as 19, okay. I am not able to reduce it. I am shivering inside.
Yes, clear? Basu sir came back after the DNA class. Okay? Yes. I know all of you are hungry and brain is not braining. Even I am very hungry. But I want all of you to tell me if translation is clear. Then I think we can go. I think I'll do one more part of this chapter, students, because we have regulation, we have lac operon, we have HGP, we have DNA fingerprinting. Okay, it will take another 2 hours for me to complete this. At least 1.5 hours to complete. Okay, throat is not throating. Okay, but so shall I stop the class here? Shall I do one more part of this? Or should I come after a break? Tell me fast. Okay, I know all of you are hungry and brain is not braining. Lunch break. I'll come back then. Chalo. Students, but I don't know how much we can finish today. Okay, regulation is easy. Lac operon also is easy. HGP and DNA fingerprinting will take some time. Okay. But again, HGP again lot for you to mug up. Yes. As you like, ma'am. Such a nice kids.
You're writing that pen now.
Yeah. So, okay, we'll take a break. Another part. Okay, chalo, we'll do it in another part. I don't have any problem because my throat is hurting. I feel if I take more strain, I will not have voice at all, okay, for this entire week. And tomorrow I have Tejas series. So, I don't want to strain my voice as well. So, we will stop here, students. It's okay. It's a big chapter. Everyone knows that we cannot finish it in one shot, okay. And we have done only 2.5 hours. We have completed up to translation, right? Okay. Yes, tomorrow Tejas series is there. My voice cannot go for Tejas series. Fine. I'll see you in next class. Yes. But before that, I have something to show all of you because many of you are asking me, ma'am, how do I get Tejas series? I want to do all of that. I will show you here. Okay. Don't go anywhere. I'm going to show you here. Just go here, students. First, see the second link. First is for MVSAT. MVSAT is happening in the offline centers up to 27th. Okay, so if you click here, you can see DNA plus NCERT Tejas book combo. Okay, and you can see that complete. So what is basically DNA? You know that most of the thing will be recorded. Okay, but again we have 1 month. November also will be completing the portions. After that you will get your all India test series and everything. So students, you will get the batch, okay, as well as you are going to get the Tejas at a price of 3499, but this is not for longer. We will stop this series, we will stop this offer very soon. Okay, so if anyone is thinking that I want to join DNA batch because at the end of the time I want my preparation to be stronger, then I would recommend you go for this because you are going to get physics, chemistry and bio books as well, and you can solve a lot of questions. Okay, and if there is any error that you spot in the book, you can immediately scan and put the error as well. So this way what happens, even you will be able to correct yourself. See, errors will be avoided as much as possible, but in case here and there errors comes, human made errors can come in any places. So that errors you can definitely tell them and the team is going to change it. Okay, so I would recommend all of you to go for this where you can get 3500. It's a good investment for book as well as for a course, and you know we are the teachers teaching there. With that we have additional teachers also who are teaching in the DNA batch. Okay, their recordings are all available, and with that you will get the Tejas book. So students, thank you so much for joining. I'm so sorry that I couldn't complete the entire class today because of my voice, and tomorrow I have Tejas series, so because of multiple classes my voice I cannot strain it anymore. Okay, so I'll see you in the next portion. Next I will tell you, mostly I will take it on Saturday. Okay, mostly I will take it on Saturday, or yes, mostly I'll take the third part on Saturday. Okay. Yes. So after Deeksha Ma'am's class, I'll confirm with Deeksha Ma'am what timing she is taking. After that, I will take this third part. Okay. Clear to all of you? So can I see your chats? So students, I can see that most of you are not giving the comments and all of it. So quickly go comment on the video and let me know how the class was. I hope NCERT every single line was clear to you. Thank you so much for joining. See you all in next class. Tomorrow Tejas series is happening. I want all of you to go from here itself and go and like the video there, okay, because last time full you gave energy. Yeah, full.
They'll not do. I'm sure them, ma'am. Where is it?
Okay, students, go to the channel, click on the channel. This is our channel, Vedantu NEET English. Okay, go to the live and you can see that breathing and exchange second part is there. Sir's principles of inheritance is there tomorrow. Go like for that video as well. See, I cannot see any likes here. Oh, go up. See, very poor.
Some chat is going on.
Yeah, some two people are talking to each other because their parents scolded them not to use WhatsApp. Okay, I don't want that. I want genuine students to go here and like this video as well. Okay, how do I close this? It's not close. It does not have an into sign. Okay, no problem. So students, can I get a lot of likes and comments on this video about how your molecular basis of inheritance is happening? Okay, thank you so much for joining. See you all in next class. See you all. Bye bye.